On behalf of the University of Illinois System, Mexican and Mexican American Students Initiative, I-MMAS, I thank all of you for your participation in the 16th International Symposium organized by the University of Illinois System, I-MMAS. I am Elvira de Mejia, a professor and the executive director of I-MMAS at the University of Illinois System. I-MMAS organized this symposium on health promotion in partnership with the National Autonomous University of Mexico, UNAM, the Faculty of Higher Studies, FES Iztacala, the University of Illinois Chicago, the University of Illinois Urbana-Champaign, and the University of Illinois Springfield. Thank you all for your generous collaboration. The objective of this symposium is to promote a multidisciplinary discussion to develop strategic collaborations among scientists from the FES Iztacala UNAM and the University of Illinois System in the area of health promotion. We will dive deeper into current health promotion research, practical strategies for interdisciplinary collaborations, and access to innovative resources and tools essential in both academic and community health settings. I thank all the authorities and representatives of different institutions for the support, especially Dr. Timothy Kalin, President of the University of Illinois System, the Ambassador Reina Torres Mendívil, Consul General of Mexico in Chicago, and Dr. Francisco José Turner Morales, Head of the Research and Postgraduate Studies, Faculty of Higher Studies, Iztacala, UNAM. On behalf of I-MMAS, I thank all the speakers for their meaningful and engaging presentations. Thank you for the exchange of knowledge and experience on the highly relevant topic of promotion of health. Thank you to the attendees for your interest in this topic that affects us all. A special thanks to Dr. Isaac Obed Perez Martinez, Department of Internationalization, FESI, Secretariat of Development of International Relations. I thank the people behind the scenes, Eric Olson, Educational Application Support Specialist at the University of Illinois System, Nancy Salinas, I-MMAS, and Mr. Aldo Mora, UNAM Chicago. Now, I am pleased to introduce the Ambassador Reina Torres Mendívil, Consul General de Mexico, Consul General of Mexico in Chicago, Dean of the Chicago Consular Corps. Ambassador Reina Torres Mendívil took office as Consul General of Mexico in Chicago on July 14, 2019. During her diplomatic career, she has held various positions in the Mexican Ministry of Foreign Affairs, including General Director for Latin America and the Caribbean, Deputy General Director for International Policy on Human Rights, and Deputy Chief of Staff of the Secretary of Foreign Relations. She was Deputy Chief of Mission in the Mexican Embassy in the Czech Republic and political liaison in the Embassy of Mexico in the United States. Ambassador Torres also served as General Director for Protection of Mexicans Abroad. Before her current position in Chicago, she served as Consul General in San Antonio, Texas. Ambassador Torres was a resident fellow at Harvard University's Whitehead Center for International Affairs and participated in the Hemispheric Security Program at the Inter-American Defense College in Washington, D.C. She graduated from UNAM, the National Autonomous University of Mexico, and holds a master's degree in International Relations from the London School of Economics. Estamos muy orgullosos y agradecidos de nuestra querida embajadora Torres Mendívil. It is an honor to have you here, Ambassador Torres Mendívil, to deliver the welcome remarks, and I'm delighted to pass the virtual microphone to you. Thank you.
Thank you so much, Doctora. Thank you so much, Doctora de Mejia. As always, it's a pleasure to participate in the events that I-MMAS organizes. I'm really humbled to see the amount of participants that you have in this symposium. That speaks of the success of the events that are part of the initiative of I-MMAS and the University of Illinois system. I am also very proud that my alma mater, UNAM, in this case, UNAM FES Iztacala, is participating. So I'm humble, as I said, to be with all of you. This morning, I want to speak a little bit of the importance and the relevance of these partnerships that we have with universities. You may think about, the consular activities or the diplomatic or consular diplomacy in different terms. I mean, many people that are not familiar with the work that we do may think of, we provide services to the community, we do passports, consular IDs, and perhaps political connections or the promotion of trade and investment. But this is one of the most novel parts of our job, to create the connections between academic institutions, between students, between people, trying to figure out how to resolve problems for the communities. And what could be more important than the health of our communities? So this is part of a really long-standing and very solid partnership between the University of Illinois system. Dr. Timothy Kailin has been instrumental in all this work to make the connections and to increase the content of this partnership with UNAM. So as I was saying, the issue that is going to be addressed in this symposium, health, is something that goes way beyond politics or what is being discussed perhaps in capitals in Washington, D.C. or Mexico City, but it's really actually one of the most relevant issues for our communities. And particularly in these days, that we hear a lot of things in the news, that we see a lot of things in the social media regarding the relations between Mexico and the United States. I just want you to focus on what is happening here today. These partnerships, these positive connections, and the long-standing partnerships is what put content in what the two countries can do together and the potential of those really constructive engagements. So once again, congratulations. Thank you to all the participants. Gracias, qué gusto saludar a todos los colegas de la FES y de la UNAM y aprovechar, hay que aprovechar estos espacios to create the opportunity to exchange good practices and to all of us learn from each other.
Now, we welcome remarks from Dr. Francisco José Torner Morales, Head of the Research and Postgraduate Studies and the Coordinator of the Water Quality and Water Contaminant Removal Process Laboratory, Faculty of Higher Studies, Iztacala, UNAM, and it is my honor to have you here, Dr. Torner Morales. Please.
Thank you. Thank you, Dr. Elvira de Mejia. Thank you very much for this organization. Thank you also to Dr. Isado Beck, which is from our internationalization office at FES Iztacala. Thank you all of the speakers that are here today. We have a great opportunity to reinforce collaborations and points of common ground on this very important issue, which is one of our main research topics in FES Iztacala, which is health promotion. Thank you, Ambassador Torres Mendívil. Thank you for your commitment to join us today. Gracias por acompañarnos y estar con nosotros. Es un gran honor. So, on behalf of our faculty head, Dr. Maria del Coro Arizmendi Arriaga, I welcome you all, speakers and attendants, to this health promotion symposium. Thank you very much to all the participants. Por mi raza, hablar el espíritu.
Good morning, everyone. I would like to say that for me it's a great honor to stand before you today. I would like to begin by expressing my gratitude to the organizers, the Illinois University, and the special organizers for this academic effort at UNAM, Thank you to Dr. Elvira de Mejia and all of your team. And well, I would like to start that mention that my name is Federico Avila Moreno. Let me see. I lead the Lung Diseases and Functional Biogenomics Laboratory called LUBIFE at Biomedicine Research Unit belonging to the FESIS Tacala. And well, recently I have a deputy director position at basic research at National Institute of Cancerology at Mexico City. And we have worked in academic terms for near of 10 years with two National Institutes of Health in Mexico City, and obviously with two postgrades, postgrad master and doctoral programs named Biological and Biomedicine Sciences, belong to the UNAM. And so I'm here today for to compelling and rapidly evolving field that I have entitled Cancer Epigenome Reprogramming for Preclinical Studies to probably identification of the actionable biomarkers in lung cancer. And to start with my presentation, who we are in LUNIFE Group. Well, we are several members of the in terms of students at pre-grade and post-grade level. For us, it's essential to acknowledge that the scientific progress is never a solitary endeavor. The work that I will presenting today is a result of a concert collaborative effort. The foundation of our work in our laboratory is based on of the several talented team based on the research, including PhD candidates, master students, and undergraduate students. And well, I cannot list every member here, but their commitment is uncertain in every piece of data that I will share with you today. First, we have academic collaborations and agreements with UNAM and INER, that's National Institute of Respiratory Diseases, that we have called INER, and INCAN, the National Institute of Cancerology, both National Institutes of Health in Mexico City. We have this agreement with UNAM, and second, we have an agreement between FESIS Tacala and Groningen University in the Netherlands. And well, based on that, we have these robust collaborations in terms of international academic collaborations. In addition, we have medical and biomedical international academic interchanges. We have very proud about our PhD students that are actively engaged in academic states with the University Medical Center for Groningen. One of these is Priscila Pineda Villegas. This is her thesis title based on lung cancer and COPD, chronic obstructive pulmonary diseases. And the other one is Octavio Trejo Villegas. Both are focused on lung cancer progression and molecular mechanism involved. And obviously, several students based on undergraduate level with FESIS Tacala, and we are very proud about your participation. Based on these collaborations, we have the honor to conduct recently an academic effort in terms of medical oncology and academic collaborations between UNAM and FESIS Tacala with this genotyping unit that we have the possibility to establish an additional medical collaboration with FESIS Tacala and INCAN. When we have several efforts to identify genomics and genetics and bioinformatic analysis to get oncological diagnostic therapies and to provide obviously cancer research in terms of basic and clinical research, and to provide results in terms of clinical diagnosis in INCA. And we have several efforts and supports from pharmacological industry, for example, AstraZeneca and Roche for this medical effort between Fesistacala and this genotyping unit. We have several efforts in terms of the comprehensive genomic profiling to identify these actionable biomarkers in terms of the solid tumors study. We have to perform several experimental process and performs in terms of the Avenue platforms. The final goal is to get a final oncological decision for the medical oncologist for our patients. And we have recently our statisticals. We have, when we use the data from the Global Cancer Observatory for 2022, the statistics observing Mexico face approximately 200,000, 10,000 new cases, and tragically over near of 85 cancer deaths. We have focused on lung cancer, and you can see that lung cancer is not the first case of incidence and mortality, but we can see that the number of mortality or the rate of mortality is near of 90% of higher mortality. Based on that, we have interested into study lung cancer mechanisms in terms of the progression disease and what is the reality for our institute. We have the main tumor incidence and mortality: breast cancer and endometrial cancer, and lung cancer and mesothelioma. We have this incidence, and we have interest into studying these kind of tumors in terms of the genomics and epigenetics mechanisms to obtain probably new actionable biomarkers to get a new schemes of therapy. And the incidence worldwide, by using this Global Cancer Observatory data, we have to get and so that lung cancer is the main the first place of terms of the incidence and mortality. And additionally, we have interest into study breast cancer also. And we know that molecular alterations frequency is focused on genetic drivers, on gene drivers by risk factors in lung cancer. The main genetic alterations, as you can see, is focused on gene activation mechanisms and cycle control. Genetic mutations on these main genes focus on the tissue transformation in terms of malignant process. And we have known in the last 10 or 15 years that epigenetic alterations based on DNA methylation, histone codes, alterations, and chromatin remodulations in terms of the topological domains that is affect in terms of the risk factors. As you can see over here, we have interest in to study also good smoke exposure in terms of the biomass, because in Mexico we have populations in terms of the economic disparity for our lifestyles in terms of these risk factors. And well, the hallmarks of cancers that affect these malignant lung progressions, as you probably know, the epigenetic reprogramming and obviously avoiding immune destruction, and obviously epithelial mesenchymal transitions, and another mechanism that is involved in this lung cancer progression. We have focus on epigenetic reprogramming as non-mutational alterations, with the epigenetics mutation profile and phenotypic plasticity. We have interest into determinate what is the main of the genetics and epigenetic alterations in terms of the histone marks, and genetic and epigenetic in terms of DNA methylation alterations. Epigenetic change in terms of the biological consequence may represent, as you can see, therapeutical targetable drivers and biomarkers in terms of these lung diseases to probably restore the homeostasis and control these malignant diseases. But probably you know that the epigenetic hallmarks and epigenome reprogramming in terms of the cancer biology is based on the balance of polycomb and trithorax. Based on that, trithorax, you can see over here, is a balance comparing with the repressive histone marks, writing by ACH2 enzyme. And in another condition, trithorax is involving the activation of these epigenetic marks, promoting overexpression of several genes. Above that, we have a description of new genetic alterations. And in the last 10 years, more or less, we have detected several alterations in the chromosome 7, in the P21.1 brand in the chromosome 7. In terms of the dysplasia and these alterations in the transition of the hyperplasia to dysplasia, and these alterations remains in the cancer in situ and cancer invasive, and we have several evidence that we have performed genome studies to detect gene expression and validations in lung cancer when we use two cohorts of patients of lung cancer derived from the INER and INCAN. We have analyzes the mRNA and protein expressions, and you can see this an increase of the copy number variations in the chromosome 7. As you can see, this is an increase of the number variations of the several genes located in the 7 P21.1 brand. And we have a validation, experimental validation. You can see MEOX2 is located in this chromosome 7, and overexpression of several lung cancer patients have a lower detection of histone codes, repressive histone codes, explaining in part that these overexpressions represents several epigenetic alterations. And you can see the overexpression of MEOX2 reflects a lower overall survival in terms of the months of life of our patients. But it's known the mechanism involved. And next, in this slide, I would like to show that MEOX2 and GLI1 as a potential novel biomarkers involved in prognosis and probably therapy response. And you can see that overexpression of MEOX2 represents a median in terms of the significant statistical terms, more around 15 months. And well, when we have low expressions, as you can see in this histological detection, we have an increase on overall survival. It's in terms of the MEOX2 expression. For GLI1, we have a beneficial approximation, because the median for low expression for GLI1 is around of 16 months, comparing with the high expression of GLI1 with 5 months. So when we have, in terms of preclinical studies, you can see that we have genetic silencing experimental determinations. When we use treatment based on cisplatin and TKIs in lung cancer, we have a diminution in terms of the tumor volume. And also when we perform genetic silencing for GLI1 in these preclinical studies. And also when we use TKIs, tyrosine kinase inhibitors, as erlotinib and afatinib. In others, we have detection and reduction in the tumor increase in terms of the lung cancer progressions using these mice models in vivo. So we have several experimental detections that we have. We have a proposed to determinants if MEOX2 is involved into control this chromatin remodeling complex. For example, we have here using chip qPCR assays increase of the MEOX2, and in this ECH2 when we use a genetic silencing. And also for these histone codes, we have detected that EGFR as a target therapy is with a diminution using these experimental models. When we use epigenomic studies using next generation sequencing, we use lung tumors in terms of solid tumors derived from the patients or for the clinical or preclinical models. We have detected that MEOX2 occupied several genetic sequences through the lung cancer genome. And we have several results in terms of the MEOX2 is occupied several genetic sequence controlling several genes in terms of the involved in lung cancer progression. When we use, we have detected that GLI1 is also controlling several genetic sequences in terms of the epigenome lung cancer tumors. And we have detected several oncogenic signaling pathways, where we have detected two probably novel biomarkers involved in these actionable biomarkers. As you can see, the PLCB1, we have detected in terms of the normal conditions in the tumor. When we use genetic silencing for GLI1, we have detected an increase of the SMARCB1 near of the transcription start site, and diminution of this negative histone mark that explain when we use this TCGA lung cancer patients cohort, that overexpression of the PLCB1 comparing with the normal adjacent tissues. And the other one, the PLCBR4, a diminution in overexpression in terms of this. This is interesting. Is very interesting for us because the overexpression of PLCB1 results in a diminution in the overall survival. And the other one, PLCB1 overexpression, is a beneficial biomarkers or biomarkers in terms of the lung cancer. Finally, when we use several co-immunoprecipitation analysis, we have detected that GLI1, SMARCB1, and BRD1 is interacting between them. But when we use silencing for SMARCB1, we have loss of this interaction. As you can see, statistical significant. And what we can see in these summary results, we have lost the interaction of BRD1 and GLI1 in terms of the probably controlling this ensemble of the non-canonical SW1 gene and controlling several oncogenetic genes involved in the malignant progression of the lung cancer. We have proposed the last year that the SWI as SNF5 complex is
1
involved
minute.
in several control of these diseases. And finally, we have some experiments in terms of the use of MIOX2 inhibitors that we use in clinical terms. I would like to thank to my colleagues from the INCAN and obviously from my laboratory in Iztacala. And I would like to thank everyone for this opportunity. This is my principal colleague in terms of personal and academic terms. And thank you so much for this opportunity, for this proposal for health promotion between Iztacala and Illinois University. Thank you.
Hi, my name is Maria Bondesen, and as you heard, I work at the University of Illinois in Springfield in the School of Integrated Sciences, Sustainability, and Public Health. First, I would like to thank Dr. De Mejia and her colleagues for inviting me to speak today, and then I'm going to jump right to the science. So the problem that I study is the problem of environmental pollution. According to the Lancet Commission on Pollution and Health, pollution is a huge environmental cause of disease and premature death in the world today. It is estimated that in 2015, it led to 9 million premature deaths, and this accounts for 16% of all deaths worldwide. After air pollution caused by combustion, exposures to chemicals is also a huge problem, and more than 140,000 industrial and agricultural chemicals have been synthesized since the 1950s. We are exposed to these chemicals through agriculture, pesticides, chemicals in electronics, flame retardants, for instance, industrial chemicals and byproducts that are released into the environment, cosmetics, drugs, which I'm not studying so much, but I'm focusing more on the environmental pollutants that contaminate food and water, and we're exposed to them through food, water, air, and ground. The challenge is that we have a huge amount of chemicals, 140,000 different chemicals that need to be assessed, and hundreds to thousands of chemicals are introduced every year. The current chemical testing relies on rodents, mainly mice and rats, and it is expensive, it is very time-consuming, it uses a lot of rodents, and because of that, it has been questioned ethically. This has led to a backlog in toxicity testing, and only a small fraction of the chemicals that we are exposed to have been fully evaluated for potential human health effects and environmental effects. This has been realized, of course, by different agencies, and in 2007, the National Research Council published this report called the Toxicity Testing in the 21st Century. In this report, they claim that we need to go from animal testing in rodents to look for more molecular mechanisms using high throughput and molecular methods and modern science to try to find a solution to this problem, thinking that prevention is more effective than treatment after you have developed an environmentally induced disease. So in my research, I use zebrafish. I think that's a very good model because it combines the time and cost efficiency and amenability for high throughput screening with cell culture models. But it is also an in vivo model. It has a conserved embryonic developmental pathway compared to rodents. It has chemical metabolism. And another advantage is that fish are egg layers, so they have their embryos develop outside the body. In addition, there's a lot of transgenic fish available, and they are shared within the zebrafish community, and they show fluorescence in specific tissues or organs or cells, thereby allowing for tissue-specific toxicity. And here we see a zebrafish embryo around 3 days old with fluorescence in the vasculature. And here is fluorescence in the yolk, in sensory neurons and the brain, motor neurons, and muscle. So by using these fluorescent fish, we can determine things that go wrong inside the fish because we can see it. So in my lab, we try to do high throughput screening. We breed a lot of adult fish together and can collect hundreds to thousands of embryos. We put these embryos in 96 well plates and then can add many different chemicals at different concentrations. Then we incubate for 1 to 4 days normally and do automated imaging. And then we generate the images that we sometimes analyze manually, which takes a lot of time, or we work with collaborators to do machine learning methods to analyze the data. We also use data generating technologies such as transcriptomics, looking at the total gene expression changes after exposure in the whole fish. We also do lipidomics and metabolomics, looking at all the lipids changed or all the metabolites changed. And we've done a little bit of single cell sequencing, looking at gene changes in each and every cell of the zebrafish body. Together with a high throughput screening, we generate a lot of data that needs to be computationally analyzed. I would like to share one example of a study we did, and this has to do with the obesity epidemic. So the pictures to the right here, you've probably seen, it's the development of the prevalence and projected adult obesity in the United States. And where we are now at 2026, approximately 40% of the US adult population is overweight and obese, and extreme or severe obesity right now is around 10 to 20% in different states. The prevalence of obesity is positively associated with diabetes, and this is also a time curve showing that people that have both obesity and diabetes in purple are increasing over time. Furthermore, obesity is more common in rural areas than in urban areas. And why can that be? That can be because of lack of health care, to some extent, socioeconomic status, limited access to healthy food, but also environmental factors such as exposure to agricultural chemicals can play a role. So we set out to test this hypothesis. We wanted to investigate if there are any agricultural chemicals that can affect lipid metabolism and obesity. And we tested 7 different herbicides and 1 insecticide. And these agricultural chemicals are highly used in the Midwest on corn and soy fields. And I'm going to focus on the last chemical here, tefluthrin, which is a pyrethroid insecticide. First, we used a cell line. This is a pre-adipocyte cell line that can be differentiated into mature adipocytes. And this differentiation can be enhanced by certain drugs, such as rosiglitazone, which we used as a positive control. And then we stain the cells with a stain called oil red O, which stains lipids. And we found that 3 of the compounds, acetochlor, metolachlor, and tefluthrin, increased the level of adipogenesis in this cell line. This can be replicated in vivo in zebrafish. So when we do this type of assay, we harvest the embryos and then we begin exposing them at 3 days post-fertilization. At 6 days, which is the normal day when the zebrafish embryos start to eat, we feed them a McDonald's diet, if you will, a high-fat diet consisting of dried egg yolk. And then at 11 days, we euthanize the embryos, fix them, and stain them with the same lipid stain. And this is what it can look like. You can see that both the control and the exposed fish accumulate a lot of lipids caused by the high-fat diet. But we can see that the exposed fish accumulate more lipids than the unexposed fish, and we can extract the stain and quantify the results. Now I'm going to go into the molecular part, which is there's a lot of data there, so I'm just going to highlight a few things. We found when we did transcriptomics, that is looking at the gene expression of the whole fish, that exposure to tefluthrin affects lipid metabolism. It affects liver steatosis, which is fatty liver. It also increases the genes that are involved in gluconeogenesis, and these are markers of insulin resistance. We worked together with Jason Tennyson at Indiana University, and he did the same experiment but in flies. And we found together with him that tefluthrin-exposed flies through the diet causes disruption of the insulin secretion signaling pathway. So to further look into the insulin signaling pathway, we stained insulin-producing cells in the in the fruit fly. The fruit fly doesn't have a pancreas. Instead, it has cells in the brain that produces insulin in response to food. So if you starve the flies, you can see that there is a lot of insulin. This is an insulin staining in the brain of the fly. If you feed them, the insulin is released. But if we expose them to tefluthrin, the insulin is still trapped in the brain of the fly. And similarly, in the zebrafish, this was done in adult zebrafish, if we expose them to tefluthrin and at the same time give them glucose or food, the glucose is not metabolized in exposed fish. So they have hyperglycemia, too much glucose in the blood. Finally, we did a lipidomics study where we looked at over 600 different lipids in the fish. And we found that exposure causes an increase of lysophospholipids, mainly LPE and LPC. What are these lipids? These lipids are membrane lipids. They sit in the cell membrane. They can be converted to phospholipids by an enzyme called LPCAT3. And my postdoc, Fahmi Mesmar, who did this study, he found that inhibitors of LPCAT3 has a relatively similar structure to the pesticide, to the insecticide tefluthrin. So we did a computational docking study and found that tefluthrin binds to LPCAT3, and thus we proposed that tefluthrin inhibits LPCAT3, and therefore you have an accumulation of LPC and LPE. Why does this matter? It has already been shown that altered LPC and LPE profiles are found in patients with metabolic syndrome and obesity, fatty liver, and patients with insulin resistance and type 2 diabetes. So to conclude, we think that small animal models such as the zebrafish can be used to efficiently identify the molecular mechanisms of chemical toxicity. And it can be used to prioritize chemicals for further testing in higher vertebrates or mammals such as rodents, and therefore we don't need as many rodents to test chemical toxicity. We find that tefluthrin acts as an obesogen by promoting adipocyte differentiation in cells and lipid accumulation in fish. Tefluthrin further acts as a diabetogen by blocking insulin release and causing insulin resistance. And part of the molecular mechanism may include an inactivation of LPCAT3. So what does this matter for us humans? It has been shown relatively recently that pyrethroid metabolites, such as tefluthrin and other pyrethroids, the metabolite levels in the blood of humans here in the US correlates to an increased risk of diabetes. It is also shown that military personnel who wears permethrin is another similar pyrethroid. Military personnel who wears permethrin impregnated uniforms have very high levels of pyrethroid metabolites in their blood. Hundreds of times, it can be up to hundreds of times higher levels of pyrethroid metabolites. And military personnel also has an increased risk for prediabetes and type 2 diabetes. So because of these epidemiological studies, we are now investigating if exposures to mouse pancreatic cells and human pancreatic islets, as well as in vivo in mice, if that causes the same effects as we've seen in fish and flies. And this is done with my collaborators at Indiana University. So with that, I just want to point out that the main work was done by Fahmi Mesmar, who was a postdoc in my lab, in collaboration with Jason Tennyson, Ken Mackey, and Gergo Sanda at Indiana University. Thank you.
Thank you for inviting me here today. I'm really happy to be here and see such a large audience as well. My talk is going to be on health promotion in the context of aging and older adults, and I will ask the audience to zoom out quite a bit from the previous two talks, which were really interesting. But my talk is going to be sort of a zoom out and focused on more population-based and epidemiologic approaches informed by the social sciences. So excited to share that with you. First, I want to talk about some important demographic and conceptual foundations. Since this is the first talk today focused on aging, I want to just point out many of you may know this already, but the world is aging rapidly, and in 2020, worldwide, it was for the first time the number of people aged 60s and older outnumbered the children under age of 5 years old. And in the next several decades, we expect that the total population over the age 60 will exceed 20%. And importantly, I think 80% of older people in the coming decades will be living in low and middle income countries. our focus here at the School of Public Health at University of Illinois Chicago is on healthy aging. And by healthy aging, we really just mean healthy living, because aging is a lifelong process. But we also mean something other than just extending the lifespan or longevity. We really are focusing on improving health and also independence and quality of life. And a real important goal of ours is this concept, is captured by this concept of compression of morbidity, whereby the goal, the public health goal here is to delay the onset of morbidity, delay the onset of dependence or illness, and not necessarily increase the lifespan, but shorten the amount of time that people are sick at the end of their life. And one area that we're really focusing on quite a bit now is dementia or cognitive decline, and reducing the risk of dementia for aging populations. Currently, about 6 million Americans have dementia, and this we think that this could double in the coming decades. And importantly, research within the United States suggests that Hispanic Americans have significantly higher risk than non-Hispanic white Americans. But very important to know is that dementia and cognitive decline in old age is modifiable. It's a modifiable risk. And what we how we think about this this modifiability is in the context of the cognitive reserve theory, which suggests that our brains have the ability to compensate for pathology and deterioration by better using the neural networks within our brain, by using them more efficiently or recruiting alternate pathways. And we've seen evidence of factors like early life education or careers that include job complexity bolstering cognitive reserve and helping people to reduce their risk for dementia, even in the face of physiological deterioration. But we're also starting to see more recently that even in middle age and early old age, maintaining a lifestyle of activities that are mentally stimulating can also bolster cognitive reserve and reduce risk for dementia. So that's the basic context of my work. And what we are really interested in my lab is focusing on, can our social lives affect our risk for dementia? So some of the work we've done has focused on social networks and social integration. And in fact, a recent study that I published with colleagues, found that for every 2 years of living alone, which is used in this context as a proxy for social isolation, we found that approximately 10% risk increase in the risk of developing dementia. This is based on a large nationwide survey. More recently, we are asking, this is with my doctoral student, Veronica Howell, we are asking the question, if social isolation is bad for cognitive health, how much social interaction is needed to avoid risk? And we have been starting to analyze data, and others have also shown that people with larger social networks have better cognitive health over time. But we're digging even deeper into that. And what we're finding is that other than having a spouse, which is very important for cognitive health, but it may not be the people you see every day that matter the most for maintaining cognitive health. We are finding interesting research that suggests that maintaining close relationships with people that you see only occasionally is very important for maintaining cognitive health. This gets at, these are people that are outside of your family probably and outside of your household, and it gets to the idea, I think it speaks to the importance of network diversity and having a constant flow of, or a regular flow of new information, new perspectives from people to maintain cognitive stimulation. So that's kind of work on social networks. Other work that we're doing now, though, and what I'll talk about for the rest of this talk, is focusing on the neighborhood context or the community context and how social lives within communities might affect risk for dementia. And this is a new area of research, and there's some new concepts that have been introduced into the literature around this. Sometimes it's referred to as cognability, where the graphic you see here is a website from a team at University of Michigan. Refers to this area as cognability. Others think of it as cognitive capital. There's a couple of references down here that you could follow to read more, but it's really all about how a neighborhood context may support cognitive health in late life for its inhabitants, and focuses on the extent to which the neighborhood has an environment that promotes physical activity, and whether it has resources for promoting cognitive stimulation and social connections. So with that in mind, we conducted a study a couple years ago that's published in the American Journal of Preventive Medicine. And the premise was that access to neighborhood resources like walkability and other forms of what we call social infrastructure may influence risk for cognitive decline among residents. But we wanted to ask, does access to these beneficial resources matter more for individuals who have limited personal resources? The idea here is that if you have limited personal resources, you might rely more on the neighborhood resources. If you have abundant personal resources, you may not rely as much on the neighborhood resources, and because you're better able to go outside the neighborhood for meeting your needs. And this is, sure enough, this is our hypothesis, is that access to these cognitive friendly neighborhood resources would be more strongly associated with cognitive health among those with limited individual socioeconomic resources compared to those with abundant socioeconomic resources. This is another way of saying this exact same thing, is that individual level socioeconomic disparities in cognitive function, which we see, which are very prominent in the United States, individual level socioeconomic disparities in cognitive function are reduced in neighborhoods or areas with greater access to cognitive friendly neighborhood resources. That's our hypothesis. To conduct this study, we used data. This is the part where you zoom out. We used data from a nationally representative panel survey of community dwelling older Americans called the Health and Retirement Study. In our study, we used the data was based on over 10,000 people, unique individuals who responded to a survey, and we merged this data with neighborhood level data at the census tract level from the American Community Survey, which is a study that's linked to our census, and then some other data from the Environmental Protection Agency. I'm going to pause here quickly to say that there's also, if you haven't heard of the Health and Retirement Study in the US, but are interested in it, there's also a sister study called the Mexican Health and Aging Study that is based on, the methodology is very similar to the Health and Retirement Study, similar measures in the survey, similar survey. And in my lab, my student, Veronica, who I showed earlier, she's very interested. Her grandparents are from Mexico, and she's very interested in doing some comparative work with the Mexican Health and Aging Study and the Health and Retirement Study. So I'm really hoping that this, me being here today, will lead to some potential collaborations for us, for anyone interested in the Mexican Health and Aging Study. For this study, we measured cognitive status with a telephone interview, a measure called the TICS. We measured individual socioeconomic status with education, household income, and wealth. Controlled for lots of other individual level factors. And the key measures at the neighborhood level, one of them was walkability, which was measured by the EPA, our Environmental Protection Agency. But it was an index focused on intersection density of the census tract, the distance to different transit stops, and also a measure of employment types. We also measured at the neighborhood level concentrated disadvantage, which was a marker of general resources for the neighborhood. And we did some complicated data analysis using hierarchical linear models, which allowed us to capture cognitive status change over time within individuals, but also compare or also look at how the neighborhood factors influence the individuals. All right, so here are the results. And what we found is that regardless of neighborhood access to neighborhood resources, regardless of what neighborhood people lived in, people with higher socioeconomic status had better cognitive health over time. That was not super surprising. We did also find that people who lived in a neighborhood that had a higher score of walkability had better individual cognitive health, and people who lived in neighborhoods with concentrated disadvantage also had better or had worse cognitive health after controlling for all the other variation. What was most interesting, though, is this last point, and this is captured. This is all of this is captured in this one graphic, but we found that individual level socioeconomic disparities in cognitive function are narrower in neighborhoods with more walkability and less concentrated disadvantage. So you can see the individual level socioeconomic pattern here by the separation of these lines. The people with higher socioeconomic status have better cognitive function throughout the entire period. The slope of this line that going upward suggests that regardless of individual socioeconomic status, people who lived in more walkable communities had better cognitive function. But what is interesting here is to see how these lines get narrower when walkability increases. Another way just to express this exact same finding is by looking at this graphic. And what this one shows is that people who live in the least walkable areas, the width of this line here shows the socioeconomic disparity, the gap in cognitive health between the lowest income and the highest income. And you can see the difference is 0.35 here. But if you walk all the way up to the most walkable community, this gap, this socioeconomic disparity, is cut in half or at least by half. So that is really interesting to us and leads us to our conclusion. And that conclusion is that we think that these findings in terms of health promotion for older adults suggest that communities can help improve older adults' cognitive health by improving things like walkability, and it can also and reduce socioeconomic disparities in cognitive health. This can be done by prioritizing pedestrian-friendly infrastructure, getting people promoting the ability for older people to get out of their houses, interact with people outside of their home, and get exercise also. So creating opportunities to get for pedestrian-friendly infrastructure that facilitate cognitive, social, and physical stimulation. Another, similarly, same result suggests that lower SES older adults who live in low resource neighborhoods experience a sort of double jeopardy with respect to cognitive health, whereby they have a baseline higher risk owing to their low SES, and they have limited opportunities for social engagement in the neighborhood that might mitigate that cognitive decline. So thank you all for listening to this. And I, like I said, I'm hoping that we can drum up some collaborations from this for anyone interested in cognitive health in older adults and this kind of population-based social epidemiologic approach.
It's a real pleasure to be here this morning with you all, and excited to talk a little bit about some of the great research that we're doing here in partnership with many other great collaborators as well. So I'm going to just share my screen here. Hopefully everyone, I'm going to put it in presentation mode and we should be good. Is everyone seeing that okay?
Yes, thank you.
Wonderful. Okay. So as Elvira mentioned, I'm the Dean of the Carle Illinois College of Medicine, which, if some of you don't know, is the world's first engineering-based medical school, which is this intersection of engineering and medicine to really think about how to solve problems for the future of health. And our students are incredible. We get students from all over the country, about 3,000 of them that come to apply for 64 slots. We have some that are from all over the US, some have advanced degrees, a lot of them are engineering majors. And we have some unique characteristics is that we now, after being only 10 years old, we're the highest in the nation in terms of the quality of our applicants, meaning they have the highest entering GPA of any other medical school in the country. And we also have the highest percent in the country of our students getting their top residency all over the US and in all kinds of specialties. So we're very proud of our students. I want to talk a little bit about some of the research we're doing. We have a lot of research in the College of Medicine that have an opportunity to collaborate with others here in Mexico as well, and looking at everything from public health and health disparities, tissue engineering, global health. We have our global consortium that several institutions in Mexico are a part of. So I'll talking a little bit about our research program. And we do have a lot of interest in partnership. We've grown quite a bit in the last 10 years in terms of our funding and some of the opportunities we have in the College of Medicine. And so with that in mind, I want to talk a little bit about tissue engineering. So tissue engineering at the University of Illinois Urbana-Champaign, you'll probably hear a couple more lectures on this today, one from the esteemed Professor Brendan Harley, who's going to talk a little bit about his amazing program. But we have a great group of collaborators across the campus. The Institute for Genomic Biology has a theme on regenerative biology and tissue engineering. We're trying to build this pipeline for tissue engineering that includes great groups of investigators, great facilities, great translational opportunities, and really moving things through this pipeline to really get into first-in-human trials. And as part of that, we're thinking about everything from biomaterials, and I'm going to talk a little bit about some of that we're working on in my lab, as well as how we're actually thinking about replacing whole organs and tissues. And this is really a collaborative effort that includes everything from how we're thinking about stem cells and new molecules to how we move those into large animal models. So you may or may not know that the University of Illinois Urbana-Champaign has an incredible large animal facility. We have a swine facility on the campus that's one of the largest in the country, where we can do large animal studies under GLP conditions at scale. And you can really start to think about doing large animal trials for moving into first-in-human studies. They have a great facility there for imaging as well and surgery. So no matter what you're thinking about from a translational aspect, there's that opportunity to partner to think about how to move new technologies into the clinic. The other thing that's new and exciting on the campus and it's in my lab now is our first CGMP facility for translational tissue engineering. And this is to really move new cellular scaffolds and cellular therapies into the clinic under GMP manufacturing conditions. And we're now excited. We're almost ready to open it. It's going to be fully completed next month, but here's some pictures. All the walls are up. They're going to be doing now some of the air quality testing and getting the final bits completed. But this has been a labor of love to get this fully operational. And inside that, we're going to have our inline automated manufacturing, basically cellular process to take stem cells and convert those into new tissues, new scaffolds, and things that can actually be implanted in a patient. So I'm going to talk a little bit about two of our regenerative medicine platforms that I specifically have been working on for thinking about the next generation of curative cellular therapies and scaffolds. And the first is the world's first implantable tissue-engineered thyroid organ created from a person's own fat cells. And this is meant to be a one-time single application fully functional cure for the disease of hypothyroidism. And this is a platform technology that really looks at how adipose-derived stem cells from a person can be transformed into a variety of different other tissue types. and we're looking at different endocrine cell types, including thyroid, parathyroid, islet cells, and other metabolic producing cells. So I'll talk a little bit about some unmet need. And so if you think about endocrine diseases as a whole, these are chronic conditions that actually impact hundreds of millions of patients worldwide. And it's a problem that's often lifelong. There is no cure. People can take a daily pill as a hormone replacement, but they have a lot of issues with symptoms, and it does require lifelong medication. And the problem is that we're trying to treat a dynamic disease with a static therapy, meaning that an endocrine disorder is a disease of a hormone balance that has a regulation between the brain and that endocrine organ. And those signals change during the day from time to time. And sometimes you need more hormone, sometimes you need less. You can think about this with your blood sugar and diabetes. You can think about this with your thyroid hormone as well. But we're treating that with a static pill that a patient takes, which gives a specific amount of a drug every day, whether you need more or less. And so obviously, when you need more or less, you're going to have symptoms. And that symptom becomes very dysfunctional for a lot of patients. Our solution is to actually create a dynamic cellular therapy that has the ability to adapt to those dynamic signals and create a functional cure that's stable and improves those symptoms and can be long-term. So the part of this platform is that we have this novel way to deliver cells. We have a scalable manufacturing process. We have immune compatibility because we're using autologous cells. So you're using your patient's own cells, giving back to them so you don't have issues with tolerance or a rejection. And it allows for enhanced vascularization, and I'll talk a little bit about that. So this hypothyroidism as a disease, affects about 300 million people around the world. In the US alone, it's about 20 million patients. And everyone gets a drug called levothyroxine, which is basically the thyroid hormone that they're on for the rest of their life. But when you think about it, if you start in your 30s and 40s, even though the drug's not very expensive, you have to take that for 30 to 40 years or more, plus taking lab tests multiple times a year, plus the symptoms that you have to deal with. It's a lot that patients have to go through over a lifetime. And the symptoms are really significant. About 30% of patients continue to have symptoms even when their labs are completely normal. And that's because of these daily fluctuations they have in hormone need. The other problem is compliance. You have to take these drugs at a specific time without food. You often have to have dose adjustments. And if you don't manage this well, you can have other severe complications such as cardiovascular issues and other chronic conditions. So what we've done is we've taken this ability to take these adipose-derived stem cells from a fat biopsy or a liposuction, so a small aspiration of fat cells. We can then identify the adipose-derived stem cells and isolate them and expand them. And then we put in a lentiviral system that basically turns those cells into a functional thyroid cell. And part of that has to do with you have to include the sodium iodide transporter, and you have to have a way for the cell to express the TSH receptor so that it can receive signals from the pituitary for thyroid stimulating hormone. And once that's in place, those cells fully differentiate into functional thyroid cells. They aggregate together into organoids, and they actually will form an organ in vitro. And we then put that into a delivery system that's a proprietary hydrogel that allows us to then inject that in the muscle. And for many decades, surgeons have been taking parts of the parathyroid or thyroid, putting it in the muscle. And amazingly, these cells work by imbibition, meaning the blood flow through the muscle lets them get oxygen, other nutrients without needing an established blood supply. And so these endocrine cells can actually stay viable for several weeks or more without a blood supply until they form their own natural blood supply, which is an amazing property that allows them to function and survive. So what we were able to show is when we put in this lentiviral system, you can see the genes being expressed. And in the middle panel, you see all of the hormones that a thyroid cell would normally make, the TSH receptor, thyroglobulin, all of these other proteins that are common in thyroid function. And our initial plan was putting these on an absorbable scaffold that could be injected. The challenge with that is that only a certain amount of cells can be loaded onto these scaffolds. And when you think about scaling it to what a human would need, we would need a large volume of scaffold or many little scaffolds to be injected, which was not a viable model. So we moved to a hydrogel where we could suspend these organoids in the hydrogel, and then we could get a high enough volume concentration to really scale it. So we did this experiment actually in a small animal rodent model just to figure out how many cells are needed to make an animal that's lacking a thyroid gland normal in their thyroid function. So these are all rodents that have had their thyroids removed. So if you look on the left side of the graph, their thyroid levels TSH levels are very high, which is a sign of profound hypothyroidism. And then we start to inject the cells at different concentrations. And when you get to about 400,000 or about 500,000 cells, you can completely normalize the thyroid function within weeks. And that stays, we followed these animals out for 18 months, which is equivalent to about 50 years in a human. And they continued to remain euthyroid without any loss of cell volume. So these cells were able to maintain senescence and continue on for a year and a half. So this is a single application, lifelong cure for a chronic disease. We also tested the cells against the antibodies for the two main causes of hypothyroidism around the world. One is Hashimoto's thyroiditis, which is an autoimmune condition, and then obviously Graves' disease, which burns out the thyroid over time. And we tested the cells against these antibodies over a period, and we're able to show that even in the presence of these antibodies, because they're coming from fat cells, they don't have the antigen that reacts to that antibody, and therefore they don't get cannibalized as the autoimmune process happens in the thyroid. So they maintain their thyroid hormone function despite these antibodies being present in the blood. And so that means that even for patients with these diseases, this would be a lifelong cure that would be sustainable, which is very exciting. So we're moving this now into our manufacturing process. We're going to be putting that on site over the next several months. And then we're going to be moving this into some large animal studies with the goal that we would be able to get into first in human studies in 2028 as the first lifelong cure for hypothyroidism. So this is a very exciting opportunity for tissue engineering and regenerative medicine. The other area I want to talk a little bit about is bone regeneration. And we have a big program at the University of Illinois. Dr. Harley is going to talk a little bit about some of his work in this space. But we are looking at how can we accelerate bone healing. And part of the problem when you think about bone fractures, especially in the setting of patients who have osteoporosis or patients who have had radiation, so where you get something called osteoradionecrosis, where the radiation damages the blood vessels so that when the bone breaks, it's really hard for it to form a normal union. And so what happens is, and some of you may recognize this gentleman, his name's Roger Ebert. He's a famous film critic from the 80s and 90s. He's actually a Champaign native, so kind of a hometown person. But what you may not know about him is toward the end of his life, he developed head and neck cancer. And as part of that treatment, they had to remove part of his jaw and replace it with a bone graft. But this was after he had had surgery and radiation. So this is putting a graft in a radiated field. When that happens, only 1 out of 5 or 20% will actually heal. So 80% of the time you get a non-union, so the bones don't grow back together. And this is what happened with him. And this was a picture on Esquire magazine about 6 months before he passed. And you can see in his jaw that the bone never healed and creates this terrible deformity. So surgeons have no good solutions for how to really get bones to heal in the setting of osteoporosis or osteoradionecrosis. So a lot of people think about using scaffolds that create bone cells growing on those scaffolds, and that's one way to think about bone regeneration. But another way that we've approached it is, what about the vascularity? The vascularity around the fracture site is critical to those bone cells coming in and forming new bone. And so what happens when you fracture a bone is that it tears the blood vessels around the capsule of the that surround the bone. And that those bleeding that happens, or the clot, contains a lot of iron. And that iron actually downregulates HIF-1 alpha and prevents new blood vessels from forming until it's cannibalized. So what we've done is we thought about, well, what if we could take an iron chelator and pull the iron out of the fracture site so that that HIF-1 alpha can stay constitutively active at a time it's needed to heal the bone. So we use deferoxamine, and what we actually were able to show is that if you use deferoxamine at the fracture site, you can actually cannibalize or chelate the iron that's around the fractures in a way that those blood vessels start to form normally. And an example of this is, if you take a radiated bone in the jaw, let's say, and you fracture it and pull it apart, which is the middle panel, only 1 out of 5 of those will heal. If you take a normal bone that hasn't been radiated and do the same thing, you get about 100% healing. But that 20% healing is a real problem. So if you put the DFO in that same spot at the time of a fracture, if it's radiated, and then you can leave it there for a long enough dwelling time that it chelates the iron, we can get that healing rate up from 20% to almost 91 to 92%, which is almost a normal bone. And you can see the burst of blood vessels that's happening with the DFO because that HIF-1 alpha remains active. So that vascularity allows that bone to heal normally. And we were able to show this actually in a first-in-human experiment with DFO, where we had a young man who had a tumor of his midface, and because of that, he had to have radiation as a child. And as you can see, when he grew up, his face was deformed in a way that it was sunken in the maxilla, and he had micrognathia of the jaw. To fix that, you actually have to fracture both the mandible and the maxilla and advance the face forwards. That's 4 fractures you're creating in a radiated field. 20% chance of healing for each one. So odds are not good for this young man. So what we did is we put in, this was done in a collaboration with Stanford, we put in catheters in both sides at the fracture site and injected DFO. Now, what was interesting is on postoperative day number 1, he actually was rolled over in bed and dislodged one of the catheters. So the other one stayed in place, and so essentially he became his own control. And what we were able to show is that the side that it got dislodged didn't quite heal as normally as the side that had the DFO, where there was perfectly normal and nice normal bone that formed. And that because that was able to form, it stabilized the jaw and he was able to heal. So it basically showed that the DFO really does work. And what we were able to do then is create a delivery system with the DFO that allows it to stay in place at the fracture site for the right amount of time so that this vascularity can really happen. The other really exciting thing around this is that if you take DFO in the delivery system and apply it to a fracture site, even in a normal bone, you can cut the amount of time it takes for that bone to create a normal, healed, biomechanically dense repair in half the time. So imagine it takes
1 minute, doctor, please.
Yes, imagine it takes 28 days for a bone to heal. Now you can get it to heal in 14 days, which allows people to get back to work, athletes to get back on the field more quickly. So you'll hear a little bit more about more of this project. Dr. Harley is doing some great work thinking about a new biomaterial for this, for different applications in craniofacial fractures. And we have a project that's going to be looking at a collagen biomaterial with the DFO to have an improved way for delivery. And so I'll stop there, but those are kind of 2 examples of how we're thinking about regenerating organs and tissues for the future. So thank you all for your attention. I'm happy to take any questions.
Thank you. Good morning. Good morning to everyone. Thank you for having me. I am so happy to be here in this symposia between Illinois University and FES Iztacala. Today, I'm going to talk about colorectal cancer. Colorectal cancer is a major global health problem. According to GloboCan in 2022, there were about 2 million cases and about 1,000 of deaths worldwide, ranking colorectal cancer third in incidence and second in mortality globally. Now, an important change is that colorectal cancer is increasing in people under 50. This suggests a birth cohort effect. Something in the environment and the lifestyle is changing, and people are exposed earlier in life. Particularly in Mexico, this trend is also a concern, especially in younger men, particularly in the 30 to 59 age group. So, what exposures are common in Mexico that could contribute to this trend? One exposure of particular interest in Mexico is the high intake of sweetened beverage and non-nutritive sweeteners. Mexico has one of the highest per capita consumption rates of soft drinks worldwide compared to other countries in Latin America. Soda is common in daily life and in celebrations, and to reduce sugar, many drinks use non-nutritive sweeteners or I-MMAS. These sweeteners give sweetness with little or not calories, and what common non-nutritive sweeteners is sucralose. Sucralose is a modified form of sucrose and is about 600 times sweeter than sugar. Importantly, as you can see in this graph, many women also consume these drinks during pregnancy and lactation. As shown in the graph, sweetened beverages are consumed across all trimesters and continue into lactation. This means exposure can happen during early development. So the key question is, can this early exposure change the gut and the immune system in the offspring? Sucralose is usually considered safe, but new human studies suggest it may have effects during pregnancy. In one study from the General Hospital of Mexico, mothers who consumed a lot of sucralose had newborns with more inflammatory cells identified by CD4 and CD14 and CD16 positive monocyte considered as inflammatory cells. And also, these newborns showed more inflammatory signals with higher IL-1 beta and TNF alpha and lower IL-10, which is anti-inflammatory. A second study, also performed in the General Hospital of Mexico, showed that mothers who consume non-nutritive sweeteners present changes in colostrum. Remember that colostrum is the first milk produced after delivery and is very important to the healthy establishment of immune response and gastrointestinal system in newborns. So these studies suggest that exposure during pregnancy may influence early immune development, but we still don't know the long-term consequences of the offspring. Based on these observations, we asked a focus question. Does perinatal sucralose exposure program susceptibility to colorectal cancer later in life? To address this, we developed a controlled experimental model. To test causality, we developed a rodent model in which dams were exposed to sucralose during gestation and lactation using concentrations aligned with regulatory safety benchmarks based on the FDA and JEFCA acceptable daily intakes, adjusted of mice, of course. After birth, the offspring were switched to a standard drinking water. They did not receive sucralose. And we then assess early outcomes, for example, body weight, colon length, intestinal permeability, and histopathological changes in the intestine. Consistent with prior reports, offspring from sucralose-exposed dam showed increased body weight gain compared with controls. In contrast, we did not detect any significant differences in colon length and in intestinal permeability at this stage in young mice and in adult mice. However, we examined the intestinal tissue architecture in more detail, and we observed clear difference in the barrier niche. We analyzed proximal, mid, and distal colon. We did not observe major histopathological difference in the medial and proximal colon. However, in the distal colon, we observed a significant reduction in creep length and a significant reduction in mucin-producing goblet cells. Importantly, these differences were evident at 3 weeks and at 8 weeks adult mice, suggesting that perinatal sucralose exposure induced long-lasting remodeling of the intestinal barrier niche into adulthood. As a reminder, goblet cells and mucins are essential for intestinal health because they form a mucus layer that limits direct contact between the epithelium and the microbiota. Next, we analyze immune populations in mesenteric lymph nodes and the intestinal lamina propria. And across tissues, we did not observe a major difference between CD8 T cells, B cells, or monocytes. However, we detected changes in CD4 compartment, including altered frequencies of CD4 positive, FOXP3 positive Treg cells, a population central to maintaining intestinal tolerance and homeostasis. In addition, we observed a more inflammatory interferon production by monocytes in offspring from sucralose-exposed dams. Together, these findings support the idea that perinatal sucralose exposure shifts the baseline intestinal environment toward reduced barrier protection, altered immune regulation consistent with a modified immune set point. So we decided to evaluate the consequence of these changes in colorectal cancer risks. For that, we use the AOM-DSS model to induce colitis-associated cancer in offspring. After induction, we evaluate tumor outcomes, and as you can see in this photograph and in this graph, offspring from sucralose-exposed dams develop a significantly higher number of tumors compared with controls. And in addition, in the F1 sucralose-exposed dams, the tumors were larger, suggesting not only increased tumor initiation, but also enhanced tumor outgrowth under inflammatory conditions. These results indicate that perinatal exposure increased vulnerability to inflammation-driven tumorigenesis. After that, we decide to perform a histopathological analysis. As you can see, this is the non-sucralose group, this is sucralose group, and we observe higher histopathological score in dams exposed to sucralose. Also, we observe a decrease in mucin producer cells, and we analyze expression of tumorigenesis associated markers like beta-catenin and phospho-STAT3, and we also observe an increase in these markers in dams from sucralose group. Interestingly, we did not observe any difference in Ki67 expression, suggesting that the enhanced tumor phenotype may be linked more to a barrier disruption, inflammatory signal, and microenvironment context than to a simple increase in proliferation. Overall, these tissue data support a more permissive inflammatory tumorigenic environment in sucralose offspring. Next, we decide to analyze the anti-tumor effector responses, and we found a significant reduction in CD8 positive, interferon gamma positive T cells in sucralose exposed dams. Remember that these cells are central to immune-mediated tumor control because interferon gamma supports cytotoxic function and promotes anti-tumor activity. We also analyze these changes by immunohistochemical analysis, and we also observe a decrease in CD8 positive cells in sucralose group. Next, we decided to analyze the regulatory compartment to understand whether immune suppression was enhanced. As you can see, when we quantify FOXP3, CD4 positive, FOXP3 positive cells, along with activation markers of regulatory cells like CD25 and CTLA4, we observe a significant expansion of activated Tregs in the sucralose group. Together with the reduction in CD8 positive, interferon positive effector cells, this pattern supports a more immunosuppressive tumor microenvironment, which likely contributes to increased tumor number and larger tumor size. Overall, our data, our partial conclusions indicate that perinatal sucralose exposure at concentrations aligned with the accepted AD induce durable changes in offspring that expand barrier dysfunction, immune regulation, and metabolism. First, we observe a barrier niche impairment, include reduced crib length, include reduced mucin production by goblet cells. This is consistent with weaker mucosal protection. Second, we detect immune remodeling consistent with altered immune homeostasis in early life. Third, offspring show increased body weight, pointing to metabolic changes that may further contribute to vulnerability. Functionally, these program changes translate into greater susceptibility to AOM-DSS-induced colitis-associated cancer, with increased tumor burden and size, more severe tissue injury, and impaired anti-tumor immunity characterized by reduced CD8 positive, interferon positive cells, and expanded activated FOXP3 positive Treg cells consistent with an immunosuppressive tumor microenvironment. As a take-home message, I want to say that perinatal sucralose exposure durably reprograms gut barrier and immune development, increasing susceptibility to inflammation-driven colorectal cancer. Finally, I want to thank especially to my PhD student, Aranza Mejia Muñoz. She has performed almost every the experiments that I have shown today. Also, I want to say thank you to all members of my laboratory and of course to the financial support and many collaborations in my institution and other institutions that have been permit the possibility of this work. Today, we are looking for some collaborations to go in deep of the mechanisms of the immune response alteration during sucralose consume. And Aranza is also interested in looking for a possibility to academic interchange, for example, in some laboratory of Illinois University. Thank you very much for your time. I'm open to your questions.
Hi, everyone. Thank you very much for the opportunity to speak with you today. I'm Jessica Madrigal, recently joined the faculty at the University of Illinois Springfield. I'm an environmental epidemiologist by training, so this is very population health focused. And I just want to acknowledge that most of the work that I'm presenting today was completed during my time at the National Cancer Institute, at the National Institutes of Health and the Division of Cancer Epidemiology and Genetics. So I just want to give a brief overview of what we know about industrial pollution and cancer risk. I think many of us have probably seen a smokestack like what you see pictured in the top right of the slide. And in environmental epidemiology, we primarily use occupational studies as the main source of information to determine if a chemical or metal is carcinogenic to humans. And for almost all of these chemicals, the cancer risk associated with lower level, long-term environmental exposure is really unclear. In the United States, there are over 700 different chemicals and metals that are emitted from industrial sources every year. And also every year, that number is actually growing. And when we think specifically about carcinogenicity, and we look at what the International Agency for Research on Cancer has decided on the weight of the evidence for a lot of these different chemicals, we actually find that there are about 100 chemicals emitted from industry in the US that are actually classified as known or probable human carcinogens by IARC. And for many of these chemicals, there's really strong evidence of carcinogenicity for certain malignancies or certain cancer sites. And until recently, some of the work that I'm going to present you, we really didn't understand or have any quantitative estimates of the amount of these chemicals that are being emitted into ambient air each year. And in the United States, we know that people who are from minoritized racial and ethnic groups, those who are experiencing poverty, and those who have low educational attainment actually experience a disproportionate burden of exposure to this type of environmental pollution. So I used the Toxics Release Inventory. This is a regulatory program from the U.S. Environmental Protection Agency. I used this to identify chemicals that are classified by the International Agency for Research on Cancer as carcinogenic to humans. So the map here basically shows all of the census tracts across the United States. And each of the dots on this map represents an industrial facility that in the year 2018 was emitting at least one known human carcinogen to the air. And when we use the GIS or the Geographic Information System to match these facilities and their addresses and their emissions to US census tracts, we can then pair it with census population and get an idea of who are the people that are living in the census tracts that are actually experiencing these emissions on a daily basis and largely involuntarily. So in 2018 alone, we saw that there were about 2,200 industrial facilities that emitted a known human carcinogen to the air. Overall, across that year, there were about 12 million pounds of these emissions across the US. And those census tracts where those emissions took place were actually home to about 7.4 million people. And I'm not going to go into details on this slide, but I just wanted to show you the difference in how this map populates when we actually expand this to probable human carcinogens. So chemicals for which we have limited human evidence, but some human evidence of carcinogenicity, but also molecular and animal evidence that these chemicals cause cancer. And when we include probable human carcinogens, not only do the pounds emitted per year to our air increase, but so do the potential population that are exposed. And just to put this into context, this is just a brief summary of the most highly emitted carcinogenic or probably carcinogenic chemicals across the United States. So chemicals that many of us recognize: formaldehyde, benzene, trichloroethylene, emitted in the millions of pounds per year across the United States. So one of my questions was really to try to evaluate how these emissions are associated with risk of cancer. In the United States, breast cancer is the most common malignancy diagnosed among women, and that's no different among the women in Mexico, where you have about 31,000 cases diagnosed in 2022. Lung cancer is the second most frequently diagnosed malignancy in the United States and the fourth most frequently diagnosed among men in Mexico. And we know that outdoor air pollution is an established carcinogen. And we also know that industrial activity, like those industrial facilities I showed you on the map, are contributing to outdoor air pollution in addition to vehicular traffic, truck traffic, things like that. And when we look specifically at risk for breast or lung cancer, what we find is that these industrial facilities are emitted known mammary carcinogens like benzene and 1,3-butadiene, as well as several chemicals that are classified as endocrine disruptors. And when we look specifically at what we know mostly from occupational studies about the development of a lung tumor, there are actually 11 known and probable human carcinogens that have been associated with lung cancer that are also emitted from these industrial facilities. And very few epidemiologic cohort studies have evaluated the risk from environmental exposure to these chemicals. So the work that I did just that. I used the US NIH AARP Diet and Health Study. This map shows highlighted in red the states from which the cohort participants were recruited from, with the dots representing the industrial facilities. And what we found is very similar to the descriptive work: very high emissions of benzene, formaldehyde, trichloroethylene, and a few other chemicals and metals. To look at risk of cancer in these cohort participants, this is largely based on using a geographic information system and plotting out where the person lived at enrollment in the cohort study, and then using the GIS to overlay where these industrial facilities are located and look at exposure based on the distance between these industrial facilities, the source of the emissions, and the person's home. So to do this, I create some distance-weighted, wind direction adjusted annual emissions metrics for a 9-year period prior to enrollment. And I just want to give you a quick overview of some of the results of what I find in relation to breast and lung cancers. So when I look specifically at these emissions and breast cancer, this is a postmenopausal cohort of women, some who develop breast cancer, some who don't, but all of whom we have their residential address. We can do this linkage and we can look at these different fixed distances around their home. Benzene is a known mammary carcinogen. And indeed, when we look at the women in the AARP cohort, we find that for those who live within 1 kilometer, so a relatively short distance, for those who live within 1 kilometer of an industrial facility that's emitting benzene, the women who had the highest industrial benzene emissions within that distance had almost 2-fold greater risk of breast cancer compared to women who didn't have any of these emissions within 1 kilometer of their home. And based on the chemical properties of benzene, we think it's going to dissipate in the air pretty quickly. So I don't actually see any association at these larger distances of 2, 5, or 10 kilometers. But the association within 1 kilometer, so very close to the home, is very strong. When I look specifically at lung cancer, these are the results for cobalt emissions near the home of the NIH AARP Diet and Health Study participants. And when we look at cobalt emissions and lung cancer, we find overall, when you treat lung cancer as a homogeneous outcome, we find that these participants have almost 20% increased risk for those who have cobalt emissions in the highest category of emissions from 5 to 10 kilometers. But when I can untangle lung cancer and actually split it up into different types, what we see is that we have the strongest association for cobalt for squamous cell carcinoma of the lung. So here is just the example of our finding at 5 kilometers, where those who had the highest amount of cobalt emissions within 5 kilometers of their home had almost 40% increased risk of squamous cell carcinoma of the lung. And in regard to lung cancer, we see similar associations for other metals like nickel, beryllium, chromium, and lead. I currently have ongoing analyses of non-Hodgkin lymphoma, kidney and bladder cancers, and ovarian and endometrial cancers that also have some interesting findings. But what I really want to talk about is the need to evaluate these associations with pooled cohorts, not only to increase the diversity of study participants. The NIH AARP Diet and Health Study is largely non-Hispanic white, but also to expand the geographic coverage of the cohort. So I'm currently working on a collaboration with a diverse set of cohorts spread across the United States. And to do this, it not only increases the coverage, but it enables this etiologic research to be done across population subgroups, including by race and ethnicity and other characteristics. So this map actually shows the coverage of the pooled cohort when I combine all of those US-based cohorts together. I actually get coverage of 29 states and about 1 million participants. And if we go across all of those states and combine all of the emissions of carcinogenic chemicals, we see that in that 9-year pre-enrollment period, there's over 700 million pounds of carcinogenic emissions emitted to the air around the residences of these participants. And when I pool them together, I get a population that is much more representative, not quite completely representative, but much more representative of the US population. So in closing, I just want to briefly touch base on continued research in industrial pollution. This pooled cohort will allow me to clarify these etiologic associations. And further, having more participants, I'll be able to use existing GWAS and epigenetic data to really identify more susceptible subgroups to this pollution and how that influences their cancer risk. I'll also have the ability to look at cancer sites that have higher incidence in some population groups, such as Hispanics and Latinos, including stomach, liver, prostate, and colorectal cancers. I also am looking at other environmental factors that may contribute to cancer risk and different disparities, including mixtures of different chemicals, and adding in things like exposure to different pesticides, radon, and different drinking water contaminants. I'm also taking into account non-chemical and social structural factors. So the beauty of having the residential address and the ability to do things in GIS is that I can add on all of these GIS-based data sets pretty easily once I have access to the cohort data. I think opportunities to extend this etiologic environmental cancer research to cohort studies in Mexico would be a great way to collaborate. This could be done for cancer and other chronic diseases, including those related to aging, like Alzheimer's disease. For GIS-based linkages to environmental data sets, although the industrial emissions that I've showed you are US-based data, these emissions are actually reported for all of North America annually. There's also land cover data that covers Mexico, and there's also the possibility for biomarker-based exposure assessment. So doing things like measuring these chemicals and metals from blood samples or serum samples, looking at perfluorinated substances and other persistent pollutants like organochlorine pesticides and other pesticides. But now being at the University of Illinois, I think one of the most important things that we learn from this etiologic research is really the need to incorporate what we're finding into education, intervention, and prevention. So at the federal government, etiologic research is really to inform regulatory policy. But in order to regulate industry, you really need to have policy that is going to be health protective. So right now, we're also seeing a need to highlight and educate people about these risk factors, as most environmental exposures are largely involuntary. You have to breathe the air, you have to drink the water. There's really a need to increase environmental and health literacy among the population. And to that end, I'm working within the UI health system to incorporate environmental health into their current health promotion programming. And to do that, we're doing education with community health workers and medical providers about risks related to the environment. So going beyond tobacco cessation to include talking about topics like air pollution, living close to industry, or having residential radon. And also working on the development of individual level interventions to protect health among the folks that live close to our university. So I just want to acknowledge all of my collaborators and funding for this work. And then I'll take questions if we have time. Thank you.
Thank you very much to the organizers for inviting me to this symposium. So I'm just gonna start talking about the importance of influenza infection. So why, one second. So why is important influenza? Well, influenza belongs to the Orthomyxoviridae family, and it's classified in 4 genera. And for health, human health, it is important genera A and B. And actually, more specifically, influenza A can be divided in different subtypes according to hemagglutinin and neuraminidase proteins. So influenza enters to the host through the binding of hemagglutinin to proteins in the epithelial cell that has residues with alpha-2,6 galactose. And it can invade epithelial cells from the upper respiratory tract, but also from the lower respiratory tract. So why influenza is still a public health problem? Well, just to have an estimation worldwide, we can have about 1 billion of total cases annually. And actually in Mexico and the USA, we can see that every year can increase not just the number of the total cases of influenza, but also the death caused by this virus. So as you can see here from the epidemiological data from the Health Council in Mexico, every year has been increased the number of deaths. And similar trend we can find in the data from the CDC in the US. So I would like to highlight that epidemiological data indicate a progressive increase in influenza-associated mortality. But what are the factors that dictate the outcome of the infection? So we can have a case where we have a resistant host and can successfully clean the pathogen, but also we can have a susceptible host that fails in clear the pathogen. So one of those factors that can influence the outcome of the infection is the host immune response. So those hosts that can eliminate the pathogen induce an antiviral response that is mostly mediated by type 1 interferons that activate the antiviral state in the host, so then can eliminate the pathogen. However, we have those severe cases in which this type 1 interferon response can fail, and then we will have an aberrant inflammatory response, which is mediated by cells like macrophages, gamma delta T cells, and importantly, neutrophils. Actually, in those patients who progress to a severe disease, we can find a massive infiltration of neutrophils in the lung. So how neutrophils know how to get to the inflamed tissue? Where the neutrophils are generated in the bone marrow, and once they are mature, they go to the peripheral blood, and then are recruited to the site of infection through different chemokine receptors such as CXCR1 or CXCR2. In the inflamed tissue, we can find the expression of their ligands like CXCL1 and CXCL2. But another important event that occurs when neutrophils arrive to the inflamed tissue is that they can upregulate additional chemokine receptors, those from the CCR family. And our interest is the receptor CCR3, which have these 4 ligands. but why CCR3 is important for us. So in our lab, we have studied several models of lung inflammation. For instance, we have analyzed the expression of CCR3 in the neutrophils that are recruited to the lung in the LPS-induced respiratory distress model, and also during influenza infection. So as you can see here, we can have like bone marrow neutrophils, they do express very low levels of CCR3. However, when neutrophils are located in the lung, at least half of them are expressing CCR3. And this is more evident in neutrophils that are located in the lung during influenza infection. So what is the point of CCR3 that is expressing on the neutrophils? So we have observed that when we in vitro stimulate neutrophils with different ligands of these receptors, such as CCL28 or CCL11, we found that actually neutrophils boost their effector functions. For instance, you can see here that neutrophils stimulated with the ligands can increase the bacterial antibacterial activity, the production of intermediate oxygen species, and also the production of neutrophil extracellular traps. All these effector mechanisms, of course, are designed to eliminate the pathogen. However, when these neutrophils overactivate, do the activation of CCR2 receptor, then that can be detrimental for the host. So our question is, what is the role of CCR3 in the induction of lung inflammation during influenza infection? To answer this question, we use a pharmacological approach. We use this compound that is a highly competitive inhibitor of CCR3 receptor. And what we did was infecting mice with influenza, then administrate the antagonist at different time points, and then analyze our mice. So we did a follow-up of the infection just by weighing the mice every day. And as you can see here, mice that are treated with the antagonist, they don't lose as much weight as the mice that are just infected with influenza. So these results indicate that blocking the receptor can protect the host during influenza infection. We also analyze the cells that are recruited to the lung during the course of the infection. As you can see here, day 4 post-infection, we have the massive recruitment of cells to the lung. However, this is reduced dramatically when we block the receptor. So what are those cells? Those cells that are recruited to the lung during influenza infection are mostly neutrophils. So as I mentioned before, neutrophils play a really important role in the pathological inflammation. However, again, when we block CCR3, you can see that there's a massive reduction on the recruitment of those cells. But other inflammatory markers like production of pro-inflammatory cytokines are also reduced. So here I'm just showing TNF-alpha, and we evaluate the expression of this pro-inflammatory cytokine in the whole tissue. So when we infect the mice with influenza, we have an increase in the expression of this cytokine. But again, if we block CCR3, there is a reduction on the expression of the cytokine. So, and then more specifically, we wanted to know whether these, specifically the neutrophils were producing this cytokine when we blocked CCR3. So again, here is the production of TNF-alpha on the neutrophils when mice are infected with influenza. And again, the few neutrophils that are recruited to the lung when we block CCR3, they are also producing less TNF-alpha. So this goes in line with our previous in vitro results, in which we found that stimulating the receptor, we boost the effector functions of the neutrophils. Are the production of those neutrophil extracellular traps, which are formed by DNA. And some neutrophil proteins like mieloperoxidase and elastase. So we evaluate the production of the presence of those nets during influenza infection. So as you can see, we have a basal signal in the control mice when they are not infected. Then when they are infected, you can see this colocalization in histone and mieloperoxidase, indicating that we have the production of those nets. But then again, we have a reduction of net production when we block CCR3. Of course, this might be explained because we have a less recruitment to the neutrophils to the lung, and therefore we will find less nets in the lungs of the mice that were treated with CCR3. So another major complication during influenza,
I think we
more or
lost.
less 25% of the neutrophils that, 25% of the patients that are infected with influenza can acquire a bacterial infection. And Streptococcus pneumoniae and Staphylococcus aureus are the most prevalent bacteria that induce a co-infection. So why does this occur? Well, this occurs because during influenza infection, we have epithelial damage and also an inflammatory response. And this environment promotes or facilitates the bacterial colonization. So far I have shown you that if we block CCR3, we have a reduction on the lung inflammation. So we hypothesize that if we block CCR3, perhaps this could prevent the acquisition of a secondary infection. So to answer this question, we infected mice with influenza, treated with the CCR3 antagonist, and then 4 days post-infection with influenza, we infected the mice with Streptococcus pneumoniae, and then 2 days after Streptococcus pneumoniae, we analyzed our mice. So intriguingly, what we found was that when mice are treated with the antagonist, they actually lose more weight. And then we found a higher bacterial burden when we blocked CCR3 and more recruitment of cells to the lung. So right now we are analyzing what are those cells and what is the actual role of CCR3 during the co-infection, because it seems to be protective if we have just a single infection with influenza, but then when the scenario changes to a co-infection, that it seems to be important for to protect the host. So finally, I would like to show that the expression of CCR3 in the neutrophils is also important or relevant in human pathology. So in this study from 2008, the authors reports that neutrophils that are recovered from the valve from patients with chronic lung diseases, they increase the expression of CCR3. And actually, our research group found during the COVID-19 pandemics, we collected peripheral neutrophils from healthy donors, patients that were hospitalized with severe or critical disease. And again, as you can see here, patients that were with severe disease, they also have an important increase on the expression of CCR3 in neutrophils from the peripheral blood. So, and this correlates with some clinical features, for instance, temperature or C-reactive protein, which is a marker for inflammation in humans, but not also in hospitalized patients. Also, we got some samples from community-acquired respiratory infections, including influenza A, influenza B, or SARS-CoV-2. Well, we pull here because we don't have many samples, but when we pull all the infections again, we found an increase of CCR3 on the neutrophils, indicating that CCR3 is upregulated on neutrophils whenever we have respiratory viral infection so far. So in summary, I show that long recruited neutrophils upregulate CCR3 during influenza infection. CCR3 activation enhances neutrophil functions, so this can result detrimental for the host. But if we block this receptor, then we limit the recruitment of those neutrophils to the lung. But seems that CCR3 protects against a secondary pneumococcal infection. And in humans, circulating neutrophils from patients with viral respiratory infection also express CCR3. So with that, I would like to thank people in the lab. This work has been done mostly by Fatima, Mariana, and David, but with the help of all the members of the lab. Our collaborators in Iztacala, Instituto de Biotecnología, and other instances, our funding. And with that, I will be happy to take any question.
Doctor, thank you so much, Dr. Damaya, for your kind invitation. It's great to be part of this amazing symposium. 16th, it's amazing. So today I will present our work on metabolic networks that impact estrogen receptor positive breast cancer therapy responses. But also I would like to mention some other research that overlaps with our previous speakers in particularly breast cancer as well as lung cancer. So we are particularly interested in estrogen receptor positive breast cancer, and overall, the type that we are interested in, the estrogen receptor positive type, is about the 60% of all the clinical cases. And if you look at the prognosis, generally, we see better prognosis in the subtype. However, when we look at the outcomes in terms of the localized versus metastasized disease, we know that the distant metastasis decreases survival, 5-year relative survival quite dramatically. And in our lab, we look at the impact of metabolism as well as targeting different, potential molecular pathways to improve the survival for those patients that have metastatic disease. And one thing that we focus on is the location. And more recently, we hear more of genome in addition to genome that would, define responses of the individual or cells to environmental cues. In fact, there was a Time article that was published several years ago that argued that the zip code was the number one predictor of the life expectancy. And they gave an example from Southside Chicago, showing that particularly in the Southside Chicago region, life expectancy was much lower compared to more affluent northern side of Chicago. There was almost 30-year difference in the life expectancy numbers. And actually, we did some studies with our colleagues at University of Illinois, Chicago, and looked at the impact of where individuals lived on lung and breast tumor biology. We published several papers, work done by these wonderful fellows, Ashley Santelis-Catiano, Hannah Heath, and Sabrina Akhtar, in collaboration with our colleagues at UIC as well as Massey Cancer Center. And we showed that zip code indeed predicted some of the worst outcomes that we see in those individuals that live in Southside Chicago. And we were able to delineate those pathways that were really upregulated in response to neighborhood factors, and that would affect actually the biology of the tumors that these individuals carried. However, today I will focus a little bit more on the tumor intrinsic effects of the location. And through the years, we showed that, in fact, the metastatic location changed the biological properties and therapy response. And within a tumor, the therapy response is not homogeneous. Yet we can use this information to design dietary or pharmacological interventions to effectively reduce the metastatic burden in at least animal models, and show relevance also in clinical specimens. And why are we thinking the metabolism, right? So the title of my talk was Metabolism. It's mainly because we are thinking of the effect of location in terms of excess, where a tumor is located or where cells are located within the tumor. It will determine their access to nutrients, oxygen, as well as blood supply. Also, it will affect the exposures. For example, if a person has tumors in their liver, they will be subjected to exposed to those metabolites that are resulting from the first pass metabolism or bacterial byproducts that might affect the prognosis. So I'll first start with the effect of the metastatic location. And this really initially pertains to different sites that we see the breast cancer metastasis. So the main site that we see is bone, and we are really good at treating bone metastatic disease. However, we are really doing a much, unfortunately, poorer job when the metastasis is to other organs, particularly to brain as well as liver. In collaboration with Dr. Maria Grosse Perdekamp at Carl Mills Breast Cancer Institute, we looked at the impact of metastatic location on survival. And indeed, those patients with liver metastasis had worse survival after breast cancer diagnosis and worse survival after metastatic diagnosis. Also, it took shorter for them to have metastasis after the primary disease. And when treated with different lines of therapy, the individuals with liver metastasis fared really worst. So overall, patients with liver metastasis has worse disease outcomes. Then in collaboration with MD Anderson Cancer Center, Dr. Debu Tirupati and the clinical trials unit there, we looked at a cohort that they had been following for almost 20 years. And we showed that essentially for those individuals that have liver metastasis, the current therapies that are normally given to women with metastatic breast cancer, including a medicine called fulvestrant, in combination with other therapies or alone, really was not effective. So in that study, we showed that patients with liver metastasis really do not respond to current single or combination therapies. And that led us to look at what actually happens in these metastatic tumors compared to primary tumors. And this is the part that's relevant to the heterogeneous therapy response throughout the tumor. In collaboration with our partners at Carl, we looked at 16 primary and metastatic breast tumors. But for this, we had to develop some approaches that would enable us to look at what happens within a tumor, within different regions of the tumor. And for that, we initially used what's called spatial transcriptomics. So what's spatial transcriptomics? Many of you might be familiar with bulk RNA sequencing. This is where we get a tumor. If you, our comparison is a fruit shake, we put everything in that tumor and we analyze and look at what's up and what's down. And really it gives us a mixture, a homogeneous mixture of everything. Then we have single cell RNA sequencing, and we compare this to a fruit bowl. We know that there are different fruits in this bowl, different flavors of cells, if you will. However, we still don't have the location information. So what spatial transcriptomics does is that it gives us that location information. Now we know where each of the fruits are located on the pie, and we can see what it means in this context. So we use this approach, but many of the analysis tools, they are actually quite burdensome to use. It would give us a lot of information that might not right away translate into clinically actionable items. So we designed what we call GIS Rota. So this is using geoinformatics system approaches to understand the location of different transcriptional patterns, if you will, in a tumor. And Dr. Madrigal mentioned some of these approaches being used in more population level, but we figured that this could be a great way to really ask where a particular pathway is activated in the tumor and what type of cells are spatially associated. So we developed this approach, and a wonderful graduate student, Jinyang Yu, in collaboration with our, collaborator, Ayman Solomon at National Center for Supercomputing Applications, we initially looked at some metastatic tumors that we have in the lab that we developed through an animal model. So when we look at these animals, treatment of the tumors with fulvestrant, this was the treatment that I highlighted that was not as effective in liver metastasis. We found that particularly in terms of estrogen response, we would see that those pathways that are important for metabolic regulation had a higher correlation when we treated them with fulvestrant, whereas some other pathways showed, some less correlations. Here's an example. So this is one of the xenograft tumors that we develop in mice. This is a liver metastatic tumor. Here we are seeing where would we normally have the estrogen response. And this is now looking at cholesterol homeostasis. And you can see that these two pathways almost perfectly overlay, especially when we look at areas where we have high expression of these pathways as well as low expression of these pathways. This is great. Our GIS approach works, but would it work in actual tumors that we would get from patients? So going back to our collaboration with Carl, we obtained 16 primary and metastatic breast tumors. We performed a lot of analysis. Jin Young's PhD thesis really mainly focused on digging through this data and really fine-tuning our approach to hopefully give future users a better way to ask these questions. Long story short, we are definitely seeing similar patterns to what we saw in our animal model. But I would like to highlight one use of this data we made that is relevant to how we can exploit this spatial information. So next, what we did was to ask this question as to were there any differences in primary versus metastatic tumor in terms of metabolic pathways. And, Jinyang Yu, who developed the GIS Rota pathway together with a wonderful previous graduate student, Ayça Mogol, looked at this. And they saw that essentially acetyl-CoA biosynthesis pathway was localized to almost to the periphery of the primary tumor, whereas in a metastatic tumor, we would see it expressed throughout. Looking more detailed in this pathway component, they identified those enzymes in this pathway that were upregulated with the drug treatment, fulvestrant treatment in that case. And then Ayça went ahead and looked at whether inhibiting all these pathways would improve responses to fulvestrant. And, they showed that essentially this particular enzyme called ACL ACSS2. It is indeed upregulated in these metastatic tumors in response to fulvestrant treatment, both at mRNA as well as protein level. And using an inhibitor of this enzyme together with fulvestrant indeed improved the tumor killing abilities of fulvestrant. And if you look at this pathway, obviously acetyl-CoA is at the center of many of these different biological pathways. But what we found was ACSS2 was particularly important for histone acetylation, as it was localized in the nucleus and it was recruited to those regions where estrogen receptor was, working and was contributing to histone acetylation. It would contribute to production of the acetyl-CoA that would be used for histone acetylation. Indeed, when we inhibited this enzyme in animals, we saw a clear decrease in the metastatic burden when we combined fulvestrant together with the inhibitor. And this actually matched very well with the tumor acetyl-CoA production, again showing the specificity of our approach. And it indeed, decreased the regulation of those genes when combined with this ACSS2 inhibitor that would be important for cell proliferation and other tumor-related pathways, and increase efficiency of fulvestrant that was more relevant to now tumor death-related pathways. Again, this exercise with ACSS2 indeed shows that we can use this information by simply looking at pathways that change between primary versus metastatic tumor. We can also ask other questions using the spatial information. For example, if there are pathways that are active in different parts of the tumor, we can co-target them together. And we did this by using a ketogenic diet that would target both the glucose metabolism as well as ketogenesis in these tumors. That was also quite effective, at least in our in vivo models. Another way is to find targets that are actually expressed throughout the tumor and target them in combination with fulvestrant, which also we showed that was quite effective in this context. And this is the molecular application of the spatial data. We also use the GIS augmented spatial biology to look at the impact of, for example, neighborhood violence and chronic stress in lung tumorigenesis. We published a paper and several others are in the pipeline for this. But more recently, we are collaborating with, again, our wonderful colleagues at University of Illinois, Chicago, and looking at the impact of neighborhood and chronic stress on maternal outcomes. So in conclusion, now using this GIS approaches, we know that the indeed, metastatic location changes biological properties, but within a tumor, the tumor therapy response is not homogeneous. And we can exploit these heterogeneity in the tumors by exploiting the spatial information we get from various if you will, big data generating approaches. With that, I would like to acknowledge our collaborators and people who actually did the work, particularly Jin Yongyu and Sabrina Akhtar, who are current students, as well as our previous students, Ashley Santelis Castellano, Hannah Heath, and Ayça Mogul. Many thanks to our funding, as well as our wonderful clinical collaborators. And thank you for listening to me. I'm happy to answer any questions that you have.
To share my results and the results of my group. Well, as you know, the genome contains both protein coding regions and a much larger fraction of non-coding RNAs that are ncRNA genes, each with distinct but complementary roles. Coding genes are transcribed into mRNAs that are translated into proteins that are corresponding to only 2% of our genome. And the 98% of our genome are related to non-coding RNAs that are housekeeping RNAs of transference, ribosomic, and regulatory, mainly microRNAs, long non-coding RNAs, etc. So the vast majority of the human genome is transcribed into non-coding RNAs. Oh, sorry. Oh, nice. Long non-coding RNAs are transcribed by RNA polymerase II, so they have recognition sites for transcription factors. In general, they are molecules averaging 10 kilobases in size, found specifically in certain cellular structures, some in the nucleus, other in the mitochondria. So we can speak of specific cellular dynamics and function. In addition, they have been shown to be expressed in a tissue-specific manner. It has been theorized that there are between 60,000 to 100,000 long-coding RNA genes. Their function are really complex. They can serve as a sponge for microRNAs. Also, they can regulate translation, and the most important function for me is those related to the modulation of the genome architecture. Speaking of hot air is a long non-coding RNA of about 2,000 nucleotides that regulate gene expression by guiding chromatin modifying complex to specific genomic loci. It plays a key role in epigenetic silencing and has been strongly implicated in cancer progression, metastasis, and poor prognosis across multiple tumor types. In this slide, we can see the structure of hot air. It has two specific domains, domain 1 and 2, and well, we are analyzing the specific role of domain 1 that is implicated with the regulation of chromatin. As you can see also, in this graph, we show how hot air is expressed in a long great kind of human tumors. As you can see, in all these cases, hot air in red, well, their expression in tumors is in red, and in normal tissues is in blue, is overexpressed respecting to their normal tissues. And also, we can see in this graph that the expression of hot air is related to specific stages. I mean that there is a overexpression in late stages of several tumors. Well, speaking of coding genes, we have created a public accessible database with both clinical and expression information for more than 100 patients with cervical cancer. Therefore, well, we have published several papers about the clinical relation with some specific pathways in cervical cancer. But in this case, in particular, we analyze the Wnt beta-catenin pathway in those tumors, I mean cervical cancer tumors. And we note that this pathway is activated and overexpressed in cervical cancer. So, So, well, we want to block this pathway with a specific drug that is ICRT14. That this drug is blocking the binding with beta-catenin and their transcription factor, that is TCF-left. So in several cell lines derived from cervical cancer, the behavior of this inhibition is really complex. We can see, for example, that in HeLa cells, when we use in this panel, when we use this drug that blocks specifically the beta-catenin pathway, well, the inhibition is not allowed, as you can see. However, the difference between HeLa and the other cell lines is precisely the overexpression of HOTR. So when we use specific inhibitors for HOTR and the use of the drug, we can block the pathway, and we were able to promote apoptosis in these cell lines that are overexpressing HOTR. So, well, in this paper, we showed that the role of HOTR was to enhance the participation of Wnt beta-catenin pathway in this kind of cells, HeLa cells. In another work, we show how HOTR can activate also the PI3K AKT and the Wnt beta-catenin pathways. Both pathways were overexpressed or overactivated when HOTR was present. And well, in this panel, we can show that HOTR is detected when we use the inhibitor. Well, HOTR is not detected when we use the inhibitor, and a specific inhibitor, BKM 120, that is an inhibitor for P3K AKT pathway. And well, when the same effect was shown when we use a specific inhibitor for HOTR. So these results can show that HOTR is activating several pathways involved in cervical cancer progression, in this case, the P3K AKT and the Wnt beta-catenin pathways. Well, and this effect is mainly, well, in another experiments, we show that this effect is mainly activated by the activation of the HIF-1 alpha gene, because HOTR is located in the promoter of HIF-1 alpha. This work was achieved by Samuel, that is a postdoctoral researcher in my lab. And well, in this sense, we are also interested in to show how HOTR is participating in the metabolic reprogramming by regulating the expression of several key enzymes. So we use several lines derived from colorectal cancer, and also, well, as you can see in human tissues, HOTR is overexpressed respecting to healthy tissues. Also, the HOTR expression is related or is increased depending on the stage of colorectal cancer. Moreover, also the overall survival depends on the expression of HOTER. Some patients that has higher levels of expression has a better or a worse prognosis in the red line. Well, respecting to HIF-1 alpha, only the protein is overexpressed in tumor respecting healthy patients of colorectal cancer, but because the mRNA is expressed at the same level in both tissues. Taking this in account, we analyzed several metabolic genes. Well, there are a lot of genes in this paper, but the important thing is that when we depleted the levels of HOTER, we were able to show that all the metabolic enzymes were also depleted. So, well, the explanation is that HOTER is activating HIF-1 alpha in this kind of tumors. So this inhibition of this depletion of HIF-1 alpha inhibits also the expression of glutaminolysis and glycolysis enzymes. This work was led by a postdoctoral researcher, also Cecilia. Well, so in this model, I showed you that that HOTER is activating the HIF-1 alpha promoter, so the expression of HIF-1 alpha increase in these cells, and therefore the expression of glutaminolic and glycolytic enzymes are also overexpressed. However, when we depleted these levels of HOTER, well, these enzymes are also depleted. Finally, as you know, several has been shown that several vitamin deficiencies has been increasingly associated with cancer risk and progression. In the case of vitamin D, has been also associated to several tumors, and we ask if HOTER could be any interaction with vitamin D. So we use molecular docking that, well, we use molecular docking to explore the entire molecular surface of the HOTER without any predefined binding site constraints. So using several algorithms, for example, a triangular natural algorithm, etc., we were able to show that HOTER has the domain 1 of HOTER has a strong ability to bind to vitamin D3, that is cholecalciferol, and we compare with a positive control that was this molecule that has been previously reported that can inhibit the HOTER function. So, well, we believe that vitamin D binds directly to the oncogenic hot air. Therefore, its apparent overexpression may be a consequence of lower vitamin D in some patients that has this increased risk to develop cancer. So, we conclude that probably vitamin D is a natural compound that can inhibit the oncogenic activity of hot air. And we are conducting more experiments to prove that. This was a hypothesis that was recently published. And finally, well, I want to acknowledge to my research group, Dr. Veronica, Dr. Eduardo López Urrutia, and Samuel Camacho, that he is, and the graduate students that are working with us. Thank you very much.
Okay. Thank you all for the kind invitation. It's been a really lovely morning seeing what people are up to. I'm going to talk a little bit about my lab's work in the area of biomaterial design and what we're thinking about in how we start assembling tissues, which I think intersects with what a lot of the conversations have been today. So I'll start with a little bit of an overview. The thing that's really animated our research group for the last 20 years is that when we look at the tissues and organs in our body, they're incredibly complex, right? They're not a uniform sack of a single cell type operating in an amorphous manner. We're dealing with structures that have a great deal of cellular heterogeneity. We have stem cells and immune cells and vascular cells and lots of other ways in which cells interact with one another. Our tissues have structure. They can be hierarchical. They can be multi-scale. They have spatial transitions that happen across them. And they change in time through development, across disease, aging. There's a lot of ways in which these structures shift and organize in time. And we've been really interested in how we build biomaterial systems. And so these might be scaffold systems like I'll talk about today. These would be essentially porous materials like kitchen sponges that cells can migrate within and interact with one another. We also make hydrogels, which would be closer to a jello, a three-dimensional structure that cells are encapsulated within. And we're interested in how to engineer these materials to mimic aspects of this heterogeneity we see in the tissues and organs of our body. Something else we're really interested in over the last few years and into the future is also starting to think more broadly about what biomaterials can be used for really in the 21st century. And what I mean by that is I'm going to have a couple of comments later as I talk about some of our work. But we're developing material for bone regeneration. You actually heard Mark Cohen earlier introduce a little bit of it as well. As part of this collaborative project, we're developing implantable materials to help regenerate bone tissue in craniofacial injuries. And if you think about it from a biomaterial design, traditionally we would think about developing a technology and assuming that it would work well for everyone, right? And that generalization really isn't appropriate. And so what we're thinking about is how do we start creating materials that better address the broad set of challenges that face our population, to address challenges related to health disparities, to think about ways in which we can provide value to a greater population. So we want to also think about ways we could use our biomaterial tools and the engineered tissues we're starting to create to understand some of the context that affects health disparities, patient variation, and the rest. And I'm going to try to give you a flavor of that work over the next few minutes. And so one of the areas that we've been engaged in for a long time is in the space of musculoskeletal tissue engineering, this idea of developing implantable materials to aid regenerative healing of defects after injury in the context of a wide range of musculoskeletal disorders. I'm going to talk about craniofacial bone repair, but we've had applications in tendon, cartilage repair, rotator cuff repair, and the like. And one of the big challenges that we're facing is that a lot of the solutions that have been proposed or worked on in laboratories haven't exactly made it to be surgical standard of care. And so the traditional tissue engineering triad is you have some sort of biomaterial with a cell and a growth factor or cytokine, and those three elements mixed together provide an avenue to create new tissues. The reality is it's much harder than that. And also it's very hard to deliver all three of those things together for patients in a clinical setting. And so we've been thinking about new ways to imagine biomaterial design as opposed to going in as a native tissue, being in an environment that, for example, can recruit cells from the surrounding wound site and provide signals for those cells to become the actuators of healing, as opposed to having to implant large numbers of cells or growth factors from outside of the body. I'll talk about some of those over the next couple slides. I do want to also acknowledge there's an exceptional set of research tools on our campus. I'm really fortunate to work in a place that values team science and multidisciplinary efforts. And so most of my work is due to large teams of faculty members working together. One of the areas you've heard about already today is some of our multidisciplinary research institutes at the University of Illinois. The Institute for Genomic Biology is one of the homes for my laboratory. I work within our regenerative biology and tissue engineering theme, where our goal is to develop new types of tissue models that could be implantable to aid healing, as you heard from Mark Cohen, our dean of the med school, and also developing platforms to study disease outside of the body. And part of that is access to an exceptional animal facility. And so some of the work that I'm talking about today uses our pig facility that Mark Cohen also introduced earlier. This allows us to do clinically relevant repair of clinical scale defects, so that we're no longer growing a little bit of bone in a mouse. We're growing back bone the size of a mouse, which is significant in the context of clinical translation, thinking about what it takes to actually get into the clinic. So I mentioned one of the big areas that we work on is craniofacial bone repair. This is a really large burden where you see congenital issues such as cleft palate. You have post-oncologic challenges where you have resection of a tumor or a radiated wound margin. And we also have trauma in the context of civilian populations. We work primarily with high energy impacts, so car accidents. We also have a lot of other surgical repair challenges that we face. The big goal that we're trying to face here is we're trying to create biomaterial implants that can fit into complex defects that vary patient to patient. No single defect is the same. That's the first order of challenge. And these defects are complex geometrically and mechanically. We then have to address the challenges that are faced in terms of the robust immune response after injury. And then we want to think about ways to really show regenerative healing, growing back native tissue. And our goal is to create an implant that can be precisely shaped quickly, like an alloplastic or non-regenerative implant, but that work in a regenerative fashion like autologous bone. Autologous bone is a great resource for reconstruction, although it's time-consuming and difficult to do. And so having a material on the shelf, so to speak, in the operating room that can be used without significant pre-surgery manipulation has a high degree of excitement that we've been working on. And so working with Mark Cohen and Matt Wheeler here at U of I and some collaborators, particularly Justine Lee at the University of California in Los Angeles, we developed a class of material that rapidly induces bone regeneration. It drives cells to be recruited from the surrounding wound site and to form new bone tissue. We've since started to use three-dimensional printing and other generative design strategies to create reinforcing structures, kind of like rebar in concrete, that allows us to create a highly porous, highly biologically relevant material out of collagen, but that is mechanically robust and can be cut and fit to shape complex defects. We've also begun to use these materials to study immune response in disease models. So having these three-dimensional porous materials that cells can live inside of becomes an opportunity in the laboratory for us to understand how stem cells from a patient and immune cells from the patient interact to shape regenerative trajectories. And we've been doing a lot of work designing collagen materials that can recruit progenitor cells and can aid the transition from an inflammatory macrophage to a more pro-healing macrophage through material design, as opposed to requiring biomolecules or drugs. We've also started to create a class of materials that are wound models. So these are three-dimensional models in the laboratory that involve a hydrogel seeded with cells from a patient that we can create a defect in and then implant our scaffold into the defect and study the process of healing in a dish. This has allowed us to accelerate a lot of our studies of how, not just what happens in the material, but at the interface between an implant and the surrounding defect margins. Mark Cohen also introduced this as a a big challenge in the context of irradiated wound margins. We can do a lot of these studies with irradiated margins in these engineered tissue models in the laboratory to understand how we create materials that aid healing rapidly in the context of irradiated defects. One other area that we're just starting to scratch the surface of in the space of regenerative medicine is donor variability. Again, a lot of the work that we do in the laboratory uses a small number of well-defined cell lines that are either purchased or have been used. And there's increasing evidence suggesting a lot of people are left out of that story. Where we get these cell sources from, how well they represent broad populations is pretty well established as that they don't. And so we've started to build a library of donor-derived adipose stem cells from a range of donors across ages and health backgrounds, and trying to understand the variability in cell activity. We know that a single implant might not work particularly well for everyone, but we'd like to understand mechanistically why that is. And so, for example, a recent study that we published, we were looking at the difference between donors who are self-identified as either female or male, and understanding the role of donor sex on some of the metrics we care about in regenerative medicine. For example, finding that the stem cells from male donors were much more metabolically active and grew faster in our biomaterials. But as it turned out, the stem cells from female donors showed higher degree of osteogenic healing. And so this left us with questions about how do we think about biomaterial design to treat a wide range of patient populations? And we don't really know how to do this yet, but we're interested in engaging broadly with scholars that are interested in thinking about developing materials not necessarily unique for every patient, but trying to understand how population variation can be accounted for in biomaterial design. Changing gears slightly, another big area in our research group isn't just designing materials that can go into a defect to help aid healing in a patient, but also how do we build tissue models to study what happens in disease outside of the body. And tissues, as we think about them broadly, they're hydrated, they're multicellular, meaning we have many different cell types, and they evolve in time through remodeling processes and aging. And we know a lot about locally how mechanical properties of the tissue might affect cells, how multicellular interactions are important. We need to develop platforms to study them. And that this idea of remodeling or how the tissues change over time is important to account for. And so we've been really interested in how do you develop biomaterial models to study that. These have largely been done in hydrogel systems that we can encapsulate cells within a defined material environment. We moved from what is state of the art a few years ago for stem cell culture, which is bathing stem cells in just a bath of cytokines and media, to thinking about creating a biomaterial models like a jello we encapsulate cells inside of. This allows us to expose the cells to define matrix conditions, pattern in growth factors, look at diffusive transport of growth factors. But most recently, what we've been really interested in how cells interact with one another, this idea that we have multiple cell types in a tissue. Directly, you think about cell-cell contact. We've also developed tools to study how paracrine signaling or how cells secrete factors that diffuse between cell types affect cell biology as well. And we've developed a toolbox of biomaterials to do so. I'm going to highlight a couple of the projects ongoing in our lab and with our collaborators because I think they're really exciting. One area is we've been pushing for a while in developing a model of the bone marrow to study hematopoiesis, which is the production of all the blood and immune cells that we require on a daily basis, 500 billion cells a day or so. This is mediated by a very rare subpopulation of hematopoietic stem cells that are the primary cell source used for a hematopoietic stem cell transplant for a range of hematology conditions. We've been thinking about strategies to be able to expand these stem cells in an artificial bone marrow so that we have access to a wider number of these cells, and there's a number of reasons for that. One, due to the limited number of available donors and some challenges related to the availability of donors for a range of ancestral backgrounds is lower than we would like in a lot of U.S. populations. And so we've been interested in how do you build models of this environment where you have rare stem cells surrounded by a vascularized bone marrow and interacting with other cell sources. One of the strategies we've been really excited about over the last few years is working closely with Andres Garcia at Georgia Tech in Atlanta. We've started to develop strategies to encapsulate cells in hydrogel droplets. So instead of creating large biomaterial structures, we can now encapsulate 1 or 2 cells inside a small hydrogel droplet control the number of cells per droplet through a range of different biomaterial processing conditions, and then pack them together to form a granular structure, very much like sand or other types of granular systems. But in this case, we can create mosaic structures with cells from different sources mixed together in defined ratios. And this has allowed us to study processes such as stem cell interaction, the role of vasculature in promoting hematopoiesis. We've also expanded this work with Simon Rogers' group here at the University of Illinois to create mechanical understanding of how these granular systems work. And the rationale behind that is that if you want to start 3D printing tissues out of granular assemblies of soft gels, we need to know a lot about the rheology of those materials. And so there's a lot of experimental and computational models that we've been really excited to engage in to try to understand how we process granular materials and how we might use them to create engineered tissues. One of the other areas that we've been really excited about in using our engineered tissue models is in the space of cancer. And we see some great examples already today. A lot of folks on both sides of this video call thinking a lot about how we understand cancer progression, cancer therapy. My lab's been engaged for quite a long time in developing platforms to study brain cancer, in particular glioblastoma, which is one of the most common and lethal forms of primary brain cancer. This is a cancer that kills you not by metastasizing to other tissues, but by spreading invasively through the brain. It has a very poor median survival and very low survival rate. The current standard of care is trying to surgically debulk or cut out the tumor, followed by chemotherapy and radiotherapy. But as you imagine, if these cells are spreading rapidly throughout the brain, cutting out the tumor is very difficult. You put a sharp surgical margin across a diffuse cellular margin. And so you're left with patients that see recurrence within a few months and almost always within a centimeter or so of the resection cavity. And so this is the second aside. I talked a little bit about the Institute for Genomic Biology and our regenerative biology theme that I'm part of. There's another exceptional research program on our campus. There's many of them, but the other one that I wanted to at least highlight is our Cancer Center at Illinois, which is a new program that has started over the last decade to bring together scholars in spaces of engineering applied to cancer, everything from imaging and data science, engineering tissue models to understanding mechanism. And so I'm really fortunate to be one of the program leaders of our cancer engineering and biological systems theme. And our goal is to develop new therapeutic targets using platforms to identify new cancer targets, to develop new compounds that hit those targets, and then go through a discover, develop, evaluate, and use platform where we're using organoid models and bioengineered systems along with veterinary oncology models to evaluate new types of therapeutic strategies to address a wide range of cancers, including brain cancer. And so this is another space where I think there's a lot of exceptional opportunities for collaboration. This research team has about 40 faculty members associated with it, working in 5 different colleges at the University of Illinois, and is, I think, a great opportunity to engage in cancer research broadly. But in terms of some of the work happening in terms of brain cancer in my lab and within the Cancer Center, we've been really interested in understanding the primary tumor of glioblastoma, again, the cancer that is spreading throughout the brain. But this tumor is resected. And so what we're really trying to understand is what happens at the margin as the cells that are leaving the tumor into the surrounding brain parenchyma, what they interact with. My lab's been interested in developing engineered models of the vascular niche, where these cancer cells tend to proliferate, form pseudopalisades, which are local spots where they induce the regression of vasculature, and you see cells in that space exposed to a diversity of matrix, cellular, and metabolic content that's difficult to study in vivo, but a great opportunity to develop tissue engineering models of the tumor margins. We also have members of our cancer center that are studying immune interactions between glioblastoma and microglia, as well as the role of radiation damage in the delivery of immunotherapies and other antibody drug conjugates to the tumor margins to aid healing. But in the context of understanding vasculature, we've been interested in using our engineered hydrogel systems to grow engineered vasculature. And so what we're seeing here is a vascular networks on the left that are formed from human microvascular endothelial cells, astrocytes and pericytes. We get these endothelial networks that form with perivascular organization of the pericytes, astrocytes attaching with their end feet, and depositing basement membrane and tight junctions. And so we were able to show that this engineered vasculature can be used as a three-dimensional model within which to study the behavior of patient-derived glioblastoma cells. And we have a series of projects to date that has shown that, like we see clinically, patient-derived glioblastoma cells will invade within these vascular networks, become tightly associated with the vessels, and cause functional and genomic signatures of regression in this hypoxic pseudopalisades that will form. We've been able to identify the factors produced by the vasculature that aid glioblastoma invasion, and been able to show that the presence of this vasculature drives increases of drug resistance, particularly through increases of a methyltransferase, which leads to DNA changes in DNA mismatch repair and resistance to the frontline therapy, which is temozolomide. So we've identified that glioblastoma cells in the tumor margins in these vascular niches actually become more resistant to the drug we use clinically right now. And this has provided a platform for us now working with our collaborators in chemistry and in molecular biology to start to develop and test new types of compounds that might target these cells that are otherwise resistant to frontline therapy. This has also led to us using our granular system. So these are microdroplets filled with glioblastoma cells. And the exciting thing about this is that once they're encapsulated in these droplets, we can use robotic handlers to move them through a wide range of high throughput screening facilities. So we can actually test in high throughput cancer cell response to a wide range of drugs. In this case, where they're not exposed to plastic, but actually exposed to a brain mimetic hydrogel system. And this allowed us to, again, accelerate the pipeline of looking at new compounds on these glioblastoma cells. One last area that's an offshoot of, again, within our Institute of Genomic Biology is starting to use vascular models to study new types of diseases. And we have a multi-theme collaboration within the Institute that's trying to create models of the endometrium and endometriosis, particularly trying to understand the role of endometrial remodeling. So we have collaborations with folks within Illinois, such as Kate Clancy, Romana Novak, and Greg Underhill, as well as folks at other institutions in the US, trying to create models of the endometrium, the lining of the uterus, to study processes of decidualization and trophoblast engagement. We've had collaborators that have also tried to use these engineered model systems to study how endometrial vasculature grows and remodels so rapidly across the menstrual cycle, identifying some novel modes of chirality of how cells are organized and how that leads to the formation of the spiral arteries we traditionally see within the endometrium, and that the matrix environment of the endometrium is what drives that. But the area that we've been most excited about over the last few years is developing a platform to study endometriosis, particularly endometriosis.
1 minute, please.
Yep, almost done. The lining of the endometrium is lesions that form on the ovaries, which are particularly difficult to treat, relatively common, and poorly understood how they form. There's very few in vivo models of endometrioma development, and we've taken a platform to use these engineered tissue models to study how the matrix environment of the ovaries affects the attachment of endometriotic cells, showing that these endometriotic cells are highly sensitive to the matrix environment of the ovaries as opposed to other tissue environments. More recently, we're creating three-dimensional models of lesions to understand how the matrix environment and exposure to phthalates influences invasion. And these models are now using kind of a unique resource, human subject-derived menstrual effluent we're collecting in order to form these lesions. So with that, I wanted to give you an idea of some of the collaborative work we're doing using biomaterial tools to develop model systems, both in the context of tissue regeneration, some areas of cancer, and more broadly, how do we build model tissues to represent the complexity of the tissues and organs we see in our bodies. With that, I want to thank you all for your attention, the funding sources, and the trainees in my lab that do the work, and happy to answer questions.
Thank you very much. I hope you can hear me. And thank you for all of you who are still remaining and standing and listening. I also want to, congratulate Elvira and the organizing committee for putting together such a great set of excellent speakers. It's my honor to provide some closing remarks about reflections on Mexican American. This is more of a broad conversation. Let me advance here. So I have three major topics. We'll talk about the Mexican American demographics, and this is primarily talking about Mexicans in the U.S., health disparities that exist and lack. It will be interesting to see that it's not always a negative. And then provide some reflections for health promotion. First of all, if we think about Mexican Americans in the U.S., they actually are almost 2/3 of all Hispanics in the U.S. And when we hear and we see statistics about Hispanics, a lot of it has to do with the impact on Mexican Americans. If you see here, Puerto Ricans is the second largest group, Cuban Americans, and then Central and South Americans make the rest. And I'll try to make sure that when I have data for Mexican Americans, I'd highlight that. And if it's about Hispanics, we know that 60% of those are primarily Mexican Americans. The reality is that right now, about 20% of the population is Hispanics. And soon, we're talking about 25%. That means 1 out of every 4 are going to be Hispanics. I'm going to show you a quick set of slides, and just to see, this is from 1980. This is the distribution of Hispanics in the U.S. You see there's a big group in the Southwest U.S. These are primarily Mexican Americans. If you look at New York area, these are primarily Puerto Ricans, now more Dominican, etc. But this is a very Puerto Rican-centric community. And if you look at Miami area, then these are primarily Cuban Americans. So there's some geographical distributions. So I'm going to quickly go through these slides. This is 1990, so you see how it's growing. And if we're here in Illinois, in Chicago, you see how the growth. This is 2000. And this is 2006. I have a slide on 2023. But I just realized I have here the map of Alaska. Alaska, usually we shrink it and we put it in a corner. Alaska is a huge, huge place. I'm going to show you the same set of slides. I want you to pay attention to the state of Alaska. So we had Hispanics in Alaska in 1980, and in 1990, and in 2000. And in 2006. Now, I bring this up. I'm from Puerto Rico. I don't think you can pay me enough to live in the most northern county of the United States. But what that means, and this is 2023, so I found the same slides. And the state of Alaska itself, right now, the population is very similar in these states that you would see at the national level. And this part here, we're talking about almost 25%, this is 20 to 35% are Hispanics. I bring this up because, like I was saying, I personally, I don't think I would go to Alaska to live and vacation. The reason you see Hispanics in Alaska is because they are the major labor force in the US. And what you see here from 1994 to 2024, we have more than double the participation of Hispanic in the labor force. At the same time, you see a reduction in the participation of non-Hispanic whites in the workforce. So they're a very, very important group in the population of the US. And for the most part, and I have some data here for Mexican Americans, for all US Hispanics combined, Puerto Ricans, Cuban Americans, and the US population. This is just to put things in perspective. So they're younger, significantly younger than the general populations. Even if you look at the different groups of Hispanic subgroups, they're the youngest. Puerto Ricans, 31, Cuban Americans, 40 years old. If you look at the percent of that have completed a bachelor's degree, so among Mexican Americans, 15%, all Hispanic, 20, 24%, Puerto Ricans, 30, 35%. So you see a gradient here in education, and this is going to come back later in a minute because I'm going to talk about physical activity. And then this is about living in poverty. Now, these are, you would say, these are social determinants of health. Your education, living in poverty, your income, that all contributes to things you can buy and behaviors that you engage. So insurance is one of those things that it has to do with, can you afford it? Do you have money to buy insurance? And what we know is that Mexican Americans have the highest rate of not having insurance. And they also report having fair or poor health, higher than all Hispanics, higher than African Americans, and significantly higher than whites. And this reflection is important because we're talking about the group that participates the highest in our labor force. We also know that they are disproportionately impacted from diabetes. And again, it's not all Hispanic subgroups have the same health profile. In this case, you'll see that Mexican Americans and Puerto Ricans have the highest rate of self-reported diabetes. And this is based data from the National Health Interview Survey, which is, have you ever been diagnosed by a doctor that you have diabetes? What we know is that half the people that have diabetes, there's another half that don't know they have diabetes and they have diabetes. But this is self-reported, and you see that Mexican American, Puerto Rican, it's lower among Cuban Americans, some of the other Hispanic subgroups. Yet, when we look at other cardiovascular risk factors, Mexican Americans have lower levels of high blood cholesterol. They have lower levels of high blood pressure. They're less likely to smoke when compared to all Hispanic blacks, especially blacks and whites. Yet they also have higher rates of obesity. So at the population level, you see these discrepancies and you try to learn more about why is that the case. At my time working at the Center for Disease Control, I had access to the Hispanic Health and Nutrition Examination Survey, and that was conducted between 1982 and 1984. Some of you probably were not born then. But back then, what we found is that despite this higher prevalence of obesity and diabetes, Mexican Americans had lower levels of hypertension than non-Hispanic whites. And it's something that it makes you think. This is very large sample size of Mexican Americans, representative of Mexican Americans in the Southwest. Subsequent, there's been NHANES and other groups that have to really confirm some of our earlier findings. And this comes when we talk about the Latino paradox, which is they have higher life expectancy when they have a exposure to things that are not good for them. And here's some of the examples. So the lower death from heart disease for Hispanic, and these are primarily Mexican Americans, lower level of cancer death, mortality, and lowered rates of infirmary. This is all Hispanic. For Mexican Americans, actually lower than 5.6. So I'm going to quickly go through physical activity because it's today's Best Buy in public health, and it touches on all the conversation we have had today: heart disease, cancer, diabetes, bone health, all these things. Physical activity is today Best Buy in public health. Cognitive brain lately. So it's not good for you. So in this case, the yellow, the green is Mexican American. So we do know that Mexican Americans are more inactive. This is no leisure time physical activity. And as we get older, we get more inactive. But I want you to pay attention to this slide of 20 to 29. These are Mexican American men. These are Mexican American women. For both groups, the prevalence of physical inactivity in the group 20 to 29 years old is higher than the prevalence of physical activity for non-Hispanic whites. So this prevalence among 70 to 79 is lower than this prevalence among Mexican American, which tells you that this is a disparity. I mean, we do know as you get older, you'll be more inactive, but the fact that these two groups very early, we have higher levels of physical inactivity that does not contribute to overall health, Remember, it's today specifying public health. Now, the question is, can you explain this by socioeconomic status? And the answer is no. They had nothing to do with socioeconomic status. Among people who do not make a lot of money, you see Mexican Americans have a higher rates of physical inactivity. Even among those who make more money, Mexican Americans have higher levels of physical inactivity. You see this for men, you see this for women, in both groups. What happened in the middle, but still in every income category, inactivity is higher. Can you explain that by education? Actually, no. Those who have 16 years of education, let's say the bachelor's degree, you see twice the level of physical inactivity among those with high income. And if you look at those with less than high school, you see higher levels of physical inactivity. This is for men, this is for women. So you see this disparity of physical inactivity that you cannot explain by education. You may ask yourself, oh, maybe it's occupation. No, even if you look at these white-collar professionals, inactivity is higher among those. These are doctors, lawyers, teachers, etc. You see twice levels of physical inactivity than you see among whites. And if you see among blue-collar workers, you also see higher levels of inactivity. If everything being equal, everybody being blue collars, you should be able to say that levels of inactivity would be the same. So this is actually an interesting concept that we see this is a behavior. You can probably substitute a couple of other behaviors and you will see there's higher levels of inactivity. This is the last slide we talked about acculturation. We did find a positive benefits of acculturation. Those who speak English at home have lower levels of physical inactivity. Those who speak Spanish at home had higher levels of physical inactivity. Those who speak both languages are in the middle, almost a dose response between acculturation and physical inactivity. So as we move forward, we have this detrimental risk factor for the health of Mexican Americans. And it's language, it's lack of health insurance, and yet there are opportunities where they actually do better. And this is the money slide. This is the distribution of the population among Hispanic. This is primarily Mexican American. This is 2010. So these groups are probably now here, and these groups are now here. So now we have among non-Hispanic whites, a large percent of the population who is older, 65 and older. And here is a group of Hispanics, primarily Mexican American, who are younger. It is to the benefits of everybody, not just Mexican Americans, also for whites, that these people are healthy, are well educated, make a lot of money, and pay a lot of taxes, because these are the things that are going to subsidize the Social Security, the Medicare, the quality of life of older adults, of all older adults. So it is imperative as a reflection that as we look at the profile of the health of Mexican Americans, that we make an effort to make sure that they do well, not just because it's a morally right thing to do, but because it's important for everybody in the US. So we talked today about health promotion, but actually there are many things that have to do, health protection and disease prevention, et cetera. But it does have an impact, especially for the Mexican American community, that all these things that we're doing, they trickle down to the Mexican and the Mexican American population. Thank you. And this is a great opportunity also for us to think about how can we exchange knowledge and collaborate with each other. How can we podamos trabajar juntos? All right. Thank you very much.
Thank you very much, Dr. Carlos Crespo, for a wonderful reflection. We appreciate your participation here. And on behalf of I-MMAS, the Illinois Mexican and Mexican American Student and President Team Kalin, I thank all the 11 speakers for their meaningful and engaging presentations. Thank you for the exchange of wonderful knowledge and experience on the highly relevant topic of health. I thank the National Autonomous University of Mexico, Facultad de Higher Studies, Iztacala, the University of Illinois Chicago, the University of Illinois Urbana-Champaign, and the University of Illinois Springfield. Thank you all for your generous collaborations, and please, if you want to open your video so we can thank you all for a wonderful, wonderful symposium. We have learned so much And it's obvious we can collaborate, right? Together, we can bring solutions to our Mexicans in Mexico, to our Mexicans in Illinois, and our Mexican American brothers and sisters here in Illinois and the US. So thank you all, and I hope we can continue collaborating in symposium, in seed funding, in mobility of students, both sides, mobility of professors. So please be in touch, and I hope that through I-MMAS and the University of Illinois system, we can do much more together. Thank you so much.